Okay, so a very warm welcome. I've been looking extremely much forward to this meeting, and actually the idea to this meeting was born almost precisely a year ago at the SCDM conference in Baltimore, where Nikin from the FDA and Dimitris Sambas from the SCDM and myself put our heads together and started talking about, wouldn't it be great if we could sit down, regulators and other stakeholders, and discuss a little bit about the challenges and the opportunities with artificial intelligence in clinical trials. And luckily, EMA supported this immediately, and we started arranging what was supposed to be a face-to-face meeting, but now has ended up being virtual like anything else these days. Okay, so for those of you who don't know me, I'm a member of the Inspectors Working Group in Europe, and a member of the electronic subgroup is currently drafting the e-guidance. And the whole rest of the e-subgroup has also been involved with this project. And together with interested parties, so from the SCDM and from eClinical Forum and UCROF, together we formed a program committee to start planning this meeting. So the agenda is really driven in the need for communication and knowledge sharing, and you'll see that as we look further into the agenda. I want to acknowledge all the persons who volunteer to do presentations. We had an enormous amount of people volunteering, and unfortunately we could not make room for everybody in the agenda. So hopefully at that point in time we'll be able to hear some more from people who volunteered to present. So being a GCP inspector myself, I just want to really welcome this opportunity to be a little bit proactive. We often do not see the advances in technology. So we don't really see these advances in technology until it's on our doorstep and we have an application for marketing authorization where tools like this have been used, and that is not really very optimal neither for sponsors nor for the regulators. So we really want to take the opportunity to start discussing the use of artificial intelligence. So things like chatbots, natural language processing, supervised and unsupervised learning, etc. We want to start discussing how it can be implemented in ways that we can trust and where the reliability of the data and the patient safety is not suffering. So let's move the slide ahead a little bit. Yes, so the disclaimer here is also just to say that, I mean, we wanted this to be an informal meeting where we can exchange viewpoints and have good discussions. This is not a meeting where the regulators are saying what our expectations are and what the guidance is, etc., but we are here to exchange knowledge. And if you can go to the next slide, I'd just like to establish a few meeting rules here. And we have it perfectly demonstrated so far how not to do it. So please keep your microphones muted when you're not speaking. And for the presenters, if you could, like I have now, turn on the video before your presentation so people can see your faces, corona hair or not, that would be great. And if you want to speak, those of you who are in the Adobe Connect room, you have at the top, you know, the raised hand icon that is located in the upper bar. If you could please use that. And if you take the floor, then briefly introduce yourself before, and also once you finalize speaking, if you can clear your status. We will not be minuting this meeting for the same reason that it's not a decision-making meeting. This is a knowledge exchange meeting. We have a few Q&A sessions planned. They are short and sweet, hopefully. And before each session, we have the Q&A session will be managed by the moderators, and the moderators will explain before each session if they plan to have a Q&A session after each presentation, or if they want to have it at the end of the session. Please again raise your hand if you would like to ask a question. And we'll take the questions in the order of the participants who raise their hands and when they appear on the screen. If we do not have the time to answer all the questions, which I believe could be likely, then we want to also mention that there will be possibilities in a future meeting. And also the presenters have agreed that you could send them additional questions regarding their presentations directly. And each of the moderators will, in their slides, have the email addresses and contact addresses of the presenters. So we do not have plans to take questions from the chat. And again, it's deliberately also because we don't want this to be a session where questions are asked to the regulators, and then whatever we write back, which was supposed to be just an exchange in opinions, has suddenly become guidance. So that's the main reason for that. So briefly on the agenda, we have now the session 1, the welcome. We have a session 2, which will be moderated by Nhi Kinh from the FDA and setting the scene. She'll present her own team. Session 3 will be Dimitra Sambat, who's the moderator for use cases for artificial intelligence in clinical research. We'll have a short break of 15 minutes, if time allows. And then after the break, we'll have a session 5 where Alan Yeomans from UCROF will have a session on risk management and validation strategies for AI. And then we'll end with session 6, which is moderated by Patrick Nadonli and Torsten Stemler from BfArM. Patrick is from SCDM as well. And that's about perceived challenges and potential approaches by regulators and industry. And this is where we hope to have some good discussions about what is needed going forward, any major gaps in the guidance you would like to see in the future, etc.
Hello, everyone. This is Nhi Kinh. I'm from the US FDA, and I'll be your moderator for this session. I would like to have Dr. Sean Cozen, Dr. Nicholas Schaltenbrand, and Dr. Joseph Geraci to turn on their web camera. It's with great pleasure that I'm introducing these 3 distinguished speakers. Dr. Sean Cozen from Janssen R&D, Johnson & Johnson, Dr. Nicholas Schaltenbrand from Vega Informatics, and Dr. Joseph Geraci from Queen's University and Netramark Corporation. As you can see from their bio, these 3 speakers have a diverse background, being a clinician scientist, mathematician, statistician. However, there is one thing very much in common is all these 3 speakers have background in subject matter expertise, background in machine learning and artificial intelligence, as well as subject matter expertise in data science. That said, let's hear from them how they would define machine learning, artificial intelligence use in clinical trials and clinical drug development. I'm handing my virtual microphone to you, Sean. And please share your screen for your presentation.
Okay, thank you. So good morning and good afternoon, everyone. So my name is Nicolas Schaltenbrand. I'm heading the clinical development informatics group at Vega. And Vega is a service provider company. I've got an echo. I don't know if someone can hear. My key focus is to support pharma companies by combining data science expertise and data management knowledge relevant to the clinical development process. And to set the scene regarding artificial intelligence and the use of it in clinical trials, I will give you some insights today of AI footprint in pharmaceutical industry. Then I will show you how we can use AI to help us in drug development process in general. And finally, I will develop the current use of AI in clinical development space. So thanks, Camelia. So that's the first slide regarding pharmaceutical areas with AI footprints. So as you all know, AI is relatively a marketing tool at the moment. It's used everywhere in a lot of industries, and AI has been increasingly being applied in clinical research. While some people believe that it's a huge opportunity, some others are skeptical, and so I think the reality is in between both of these extremes. And it is clear that artificial intelligence tools are at the moment providing new opportunities for the pharmaceutical companies. I have reviewed the sector. I reviewed the use of AI in pharmaceutical industry, and this slide I identify 5 main, I would say, areas in the pharmaceutical sector where we are using AI. Starting for the top left, number 1, it's the drug discovery and development. In drug discovery, it's mainly used for the compound screening, for the affinity and the toxicity. And also in development, I will develop more data in the following slide that I will develop later, the way we can speed up clinical trials using AI tools. Point number 2 is operational optimization. So artificial intelligence is used a lot of processes to speed up them, to automate it as much as possible, what we do in clinical trials. At the same time, it helps us also to manage interaction with patients, like chatbots and things like that. Number 3 is a personalized medicine. And so what the aim is to design treatment by cross-referencing patient treatment and outcomes. And we use AI at the moment in this area. In gene editing, we've got several applications of AI at the moment for insertion and deletion of predictions. That's a topic key in gene editing at the moment. And finally, number 5 is the disease diagnosis. So differentiation between healthy and disease conditions, it's also an area where AI is used quite a lot to support, to help physicians in their diagnosis process. Camelia, can you make the next slide, please? So if you go down now to the key question, so can AI technologies help us in drug development? So I try to summarize the processes from the R&D. So on the left part of the slide, you've got the research, drug discovery, preclinical, the clinical trials as its own is in the middle of the slide, and then the regulatory and the post-marketing projects. And I try to give you in each of these space some idea where AI is helping us today to perform some activities. So if I'm starting from the left part, AI can help us today to identify drug targets, to predict drug properties like drug toxicity. It helps us also to find good molecules from data library and to suggest some chemical modifications. At the same time, it helps also a lot of pharma industry at the moment to identify the right candidates for repurposing of drug. That's for the drug discovery and preclinical. So if I go to the middle of this slide, which is the key focus of today, clinical trials. So I listed 5 key topics, but I've got more in the later slide. The first one is regarding improving the clinical trial design and also to modify the protocol, to optimize, I would say, the protocol. The second one is to be more efficient in selecting the site, the country, and the patients for the optimizing of the recruitment. During the clinical trial phase, we still have a lot of tools and processes in place, but with AI, we can optimize real-time analytics and I would say real-time data monitoring of the clinical trials. At the same time, AI is starting to give us some insight to help us at the moment to manage correctly remote patient monitoring. And during this COVID situation, it has been very key to try to find ways to efficiently manage and drive correctly the site and the patients, even if we are not allowed to go to the site for managing physical visits. And the last point in the middle of the slide, the patient adherence, patient compliance, patient engagement during the clinical trials. That's also areas where artificial intelligence tools can help a lot. If you go to the right part of the slide, so you've got more the NDA submission and the marketing and post authorization studies. We still have a lot of AI tools used at the moment in this area. It's starting to identification of adverse events. We've got also some pharmacovigilance tools running using AI tools. We perform real-world data analysis using AI tools, especially on the electronic health record. We can make market analysis. We can also help the supply chain and to perform better integrity of the supply chain process for drug discovery. And all in all, in the bottom of this slide, it's also the interoperability between data. And it's quite important that we use effectively this data, but also we are not managing them into silos. And it could be done using, I would say, some principle we've got in the FAIR data, and also using some standards that we've got with the CDISC standard in clinical development. Moving to the next slide, Camelia, thanks. Next, just because it's an animation. I just want to focus on the key use of AI in clinical trials. I will start with the study design, and I will give you in this slide the main part of my presentation. So study design, at the moment we are leveraging a lot of data sets of healthcare, and we use AI tools to assess and select the optimal primary and secondary endpoints in study design. So it helps a lot to ensure that we've got the right data and the right protocol in place, and we try to optimize the study design by selecting also the right country and the right strategy for the enrollment of patients. Next, Camelia. So we are using also, at the moment, protocol designers are using a lot of real-world data, and they can combine and expand their knowledge with the use of real-world data to optimize the eligibility criteria, to understand also which patients are available, how we can manage them, and also to engage directly for active patient insights. So it's a combination of real-world data to feed the study design. Next, the opportunity will extend, and we will continue to extend that with the adoption of a Chancellor-Rate Common Protocol template, and the opportunity of using AI-driven increased automation downstream in the protocol. Next, adaptive design is part of this bullet point also. So many adaptive designs can use today AI techniques, artificial intelligence techniques, to enroll patients in trials, to profile them using the DNA sequencing, proteomics, metabolomics data. And they can use also real-world data for matching drugs considered in the trial to the pathologies and identify them from the profiling. So it's very a combination of real-world data and knowledge of protocol designer to improve this study design phase. Next, then I move to the patient enrollment. So it's more classical way. You've seen that already, I guess, in several projects when we started the AI a couple of years ago. But it's mainly to mitigate the risk of patient enrollment by identifying and selecting the right country, the right site, and the right patient, and to increase, I would say, the recruitment rate and to decrease the dropout rate of patients in trials. Next, data monitoring. So AI is playing a huge role there, and we are starting to monitor in real-time data. We are starting to develop a lot of analytics techniques to try to identify quality problems during the course of the trial. So data monitoring is currently using automation, action decision frameworks, and so on for improving the clinical trial process. Next one. Medical imaging. So it's also a topic very important in diagnosis, but also in clinical trials. At the moment, a lot of machine learning techniques are used, and it becomes a driving force for image diagnostic. So we are using pattern recognition, segmentation techniques, and medical images to enable them to a better diagnosis, to track of disease and disease progression, and to generate algorithms for detecting any abnormality on medical images. So AI is widely used at the moment in medical imaging area. Next. And then pharmacovigilance, I mentioned that we've got already a lot of tools using AI technology for the extraction of the right information and to make automation of pharmacovigilance. Next one, eTMF. So it's a process you may know, that's submission of the dossier. So it can be automatically collected at the moment and classify this information. And that's the reason we try to extract all of the metadata from this document using AI techniques and can run from fruitful analysis. So it helps a lot, I would say, to try to organize and to manage this type of activities. And the last one, next, it's electronic health record. So I mentioned already that we were taking a lot of data from electronic health record at the moment to try to predict hospital readmissions to patient risk level. You've got heart failure predictions, mortality, at-home fall risk, and patient deterioration, and so on. So you've got a lot of application using extracting data from electronic health record and applying artificial intelligence tools for that. Move to the next slide. So that's the summary. So it's just to give you more or less the same topic. So to summarize my talk, the current use of AI in clinical trials is relatively a big one. And the next session managed by Dimitris will show you some very interesting cases. But all in all, to set the scene and to give you an overview in clinical trial process, AI is already there. It improves the trial design at the moment. It helps us to manage the protocol design. It helps us to identify the right patient for a trial. It helps us also to improve the drug adherence. You've got some funny things like facial image recognition to detect the patient and to ensure the patient is taking the pills and so on. So they're very good, interesting tools for drug adherence. We are using AI for real-time monitoring and analytics. And we are also using AI for medical image segmentation and classification, auto classification.
Hi, this is the personal mailbox of Dr. Joseph Taraci from Netramark. Please feel free to leave any business or personal messages here.
And finally, we can
After leaving a message, you can hang up or press pound for more options.
I don't want to leave a message to Mr. Joe. And we can, at the end, we can also in the future combine all the data coming from wearables data and combination with patient clinical history and omic profile. And that's the main point of the use of AI at the moment. And we've got a lot of initiative at the moment in the pharmaceutical community to develop new tools in this aspect. Next one, and it is my last slide. So I just want to conclude this overview, and I hope it's helpful for you to see that AI is certainly a nice tool.
Mailbox of Dr. Joseph Taraci.
I would like to address also some challenges.
Please feel free to leave any business or personal messages here. Thank you.
I would like to address also some challenges using AI in a clinical trial. So first is the limitation of deep learning models to generalize to complex medical data sets. So it's nice, and we've got a lot of proof of concepts, but we've got also some, I would say, difficulty to generalize the use of deep learning models to very complex medical data sets. That's the first challenges we've got. The second one, it's so when we will discuss supervised learning and supervised learning later on, but in the supervised learning mode, you need to have a labeled data set and to train as a network. And it needs a huge volume of data with a labeled data set. And it's not always possible, even if we've got a huge volume of patients, sometimes in some studies, it's quite sometimes difficult to address these labeled data sets stories. The third point is the validation. So we are moving now to a continuous learning algorithm. So the the algorithm is learning over time with the new data coming in, but we need to validate this algorithm, and we need to integrate this algorithm in the existing clinical trial data flow. And that's clearly a challenge also at this level at the moment. And the last one, which is quite interesting also.
Of Dr. Joseph Taraci from
the last one is regarding, finally, we need to have a clear strategy and regulatory framework for dealing with these AI tools. With the relevant ethics issues we've got, we have a patient privacy issue. We need to retain anonymity of the data. We need to secure this data, and at the end, we need also to de-risk the use of AI and machine learning tools for clinical prediction and decision support in healthcare. So that's the four main, I would say, challenges we are facing at the moment, and we try to address. Thanks for listening to me, and I try to cover and to set the scene regarding the use of AI in clinical trials. I'm fully available via email or LinkedIn if you have any further questions, and I'm pleased to hand over back to Sharon, I guess if you solve your microphone problem.
Fantastic. So it's great to be here, and hopefully we're not going to have any technical issues. My intent with this presentation is to provide a general overview of the use of AI in clinical trials and clinical development. My talk is non-technical, and I'm going to touch on some key features of AI and how we're using AI and thinking about using AI in research and development from a therapeutic development and biomedical research perspective. First, I'd like to underscore the fact that in recent years, in the past decade or so, we've made tremendous progress in therapeutic development. So this slide shows that in non-small cell lung cancer, this slide shows that in non-small cell lung cancer, we've been able to change the natural history of the disease with the therapeutics that have been approved since the early 2000s, thanks to the genomic revolution and our ability to be able to analyze the massive data that's emerging out of genomic pipelines. So if you look at the top panel, it shows overall survival in non-small cell lung cancer since the early 2000s. And you can see that with each targeted therapy, our new therapies are really making a difference. The bottom panel is the same data, however expressed in terms of number needed to benefit, meaning how many patients do we have to treat, in this case with advanced non-small cell lung cancer, for one patient to benefit. And I think this puts the progress we've made in recent years into a more patient-centric perspective. So if we look at the early 2000s in non-small cell lung cancer, the number needed to benefit was about 8, meaning we have to treat 8 patients with chemotherapy to have one patient that benefits. And if you look at where we are now, we have made tremendous progress, and our targeted therapies, the number needed to benefit for these therapies is closer to 2, meaning we treat 2 patients to have one patient that can potentially benefit. And that's tremendous progress. And a lot of that progress has come because of the genomic revolution and because of the way that we're now able to analyze data. And some people call this being as part of the social contract that biopharma has with society, meaning that there are initial investments by biopharma, government foundations, and academia that go into basic science research. And when these compounds enter the clinic, that's when clinical development begins. Of course, this has been a very complicated area because, as we know, the majority of compounds that are discovered in the lab, unfortunately, are not successful in the clinic. And some people call that the valley of death. And in fact, one of the main uses of AI is to de-risk clinical development, to first optimize the compounds and our predictive ability to bring these compounds to the clinic to increase the chances of success in clinical trials. And also, there are various ways that we're using AI to, as it was mentioned earlier, to identify patients, to accommodate patient recruitment, and to identify populations of patients that can benefit from therapies. And all of that, in a way, one way to think of it is that it's to de-risk development so we can continue to bring safe and effective therapies to areas of high unmet need. And the reason that we can now leverage AI, AI is nothing new. A lot of, in fact, the theoretical foundation of what is being used today was developed back in the 60s. But the reason that AI is finally showing a lot of promise in biomedicine is because of the fact that we have a lot of data right now. And the whole concept of big data that has captured everyone's imagination is real. And this slide just shows that big data in recent years has been on the cover of both mainstream and also peer-reviewed publications. And this data is really the ammunition that's driving a lot of the work in artificial intelligence forward. And of course, coupled with that is our increased computational capacity. And this slide just shows the exponential growth in the volume of genomic data. And in fact, one of the main reasons that we're using AI in discovery and in the preclinical environment is because the amount of data that's now being generated from genomic pipelines and other big data pipelines is really exceeding traditional bioinformatics capabilities to leverage and extract maximal value from these data assets. And when we look at the data that we're now collecting, it is pretty complex. It's not just about the genome, which was on the last slide. We now are looking at the RNA, we're looking at the proteome, we're looking at metabolites. More recently, we've recognized that the microbiome, the gut microbes, have an influence on disease initiation and maintenance and how people respond to therapies. And there's this evolving concept of exposome, that is the environmental influences on how patients respond to therapies. And these are truly complex data assets and data assets that are very unique. And when it comes to machine learning methods, these methods are one of the few ways where we can actually dissect out and tease out signal from the noise from these very complex data assets. And so another way to think about this is a very interesting paper that came out in 2017, which basically talked about this growth in biomedical big data, which are data points around a patient. And it really started to talk about the fact that the more we can analyze the data around the patient, the more we can be holistic about what actually influences the experience of the patient. And in fact, big data and AI is a way of putting the patient back together, because in recent years, we've been kind of reductionist in the way we look at a patient. For example, I'm a thoracic oncologist. That's a hyper-specialized field. And the reason that oncology was fragmented because of the fact there's so much information that we have to memorize. But now if we let AI and machine learning and technology streamline the technical aspects of data analysis and what we are now memorizing, then we can start to think about the patient quantitatively in a holistic fashion. And that exposes new opportunities for drug development. And some people argue that we are in fact nearing the limits of biomedical reductionism. And one way to look at that, despite the first slide that I showed, the amazing progress that we've had in non-small cell lung cancer, here are the single genes that have been used to develop highly effective therapies. So in 2010, these are the single genes that we knew about. 2015, on the right side, those are the single genes that we discovered. And this is what some people call a rather reductionist way, where we're just looking at one gene. We're not looking at the exposome or the proteome, this holistic milieu that really influences health and disease. So if you look from 2010 to 2015, we didn't really discover any new meaningful single driver mutations. And here's where we are in 2020, where the gray rows are the only FDA-approved products right now on the market using this what some people call the reductionist method. And in fact, you can quantify this reductionist way of looking at data and advancing therapeutic development. Here's an exercise that we did where we looked at several drug targets. I'm not going to go through the details in the interest of time, but what this shows is that given the data that we're collecting in most clinical trials and the analytical methods that we're applying, we're as accurate as it can be. In fact, so which means we're as predictive as we can be. If we want to be more predictive, if we want to develop more bespoke solutions for patients, we really need to venture into the world of AI and ML. And again, I'm going to skip these slides in the interest of time. And one way to think about AI and ML in clinical development and clinical trials And this goes back to the first slide that I showed. It's all about being more precise. It's all about tailoring treatments to the needs of individual patients. So if you look at my slide when I showed the overall survival for patients in non-small cell lung cancer with chemotherapy, that was when that we were rather imprecise, meaning after we saw a patient with a thoracic mass, we looked at a microscope and we classified using histology. That was really the best we could do. And then we gave chemotherapy. So we were treating non-small cell lung cancer at the population level. So that was the 1990s chemotherapy. More recently, with the one dream, one drug kind of paradigm that I showed, that's where targeted therapies came in. And that's where we're now tailoring treatments based on same basically patients, non-small cell lung cancer, but we're reclassifying them into smaller groups and developing therapies that are more effective. And today we're in the era of, we're getting closer to the N of 1. And cell therapy in oncology is an example where all the therapies are designed, in fact, for one individual. And the amount of data that is required for analysis when one goes from a population-based, group-based approach to closer to the N of 1 is exponentially greater. And these data assets, again, exceed our ability using traditional bioinformatics techniques to really make sense of the data. However, if precision medicine continues to be the goal, we need to take maximal advantage of AI and machine learning to just make sense of the multimodal data that we're collecting today. And this is very critical because the way that we're still, in a way, looking at patients and reporting the results of clinical trials, for example, we report the results of clinical trials in aggregate, as average treatment effects. So on the bottom panel here, my intent here was to show the results of all traditional clinical trials on one slide. And that's what we have, that you can have a normally distributed population on the left side or skewed to the left or the right. And we calculate an average treatment effect. And that is the basis of how we report and how we conduct clinical trials. Where, application of AI and machine learning is allowing us to have a much higher resolution, because when you think about it, an average or median treatment effect is not very helpful to the clinician at the point of routine care, nor the patient, because it's an abstract hypothetical concept. There is no average median patient. In fact, mathematically speaking, every single human being, every single patient is an outlier to that average, because again, the average is a statistical hypothetical concept. So the way that we're using AI is really to get closer to the N of 1, to experience of the individual. And that is really a continuation of our quest for precision medicine. And the way that we're doing it is really we're reclassifying disease, just how we reclassified non-small cell lung cancer with single genomic drivers. AI is allowing us to reclassify disease. I'm not going to go through the details here about the history of disease classification, but what's important here is that we have been reclassifying diseases throughout history. And in fact, what we know about oncology, which is my field, it is really based on the WHO histological classifications back in the 60s. Before that, it was mainly anatomical, and now it's become molecular. And that molecular classification is where AI comes in. In fact, if you look at the approval back in 2017 at the FDA for tissue agnostic indication, basically pembrolizumab, which is a checkpoint inhibitor, was approved agnostic to anatomy or histology. And that was the first approval that essentially was based on the mechanism of disease, meaning if a patient has this genomic signature, it doesn't matter what the anatomy is, because those were just based on the reason I call something lung cancer and call another thing colon cancer is because cancer patients should be treated by surgeons. And they had an anatomical approach to categorizing disease. Now with molecular profiling with AI, we can actually mechanistically approach disease and start to shift away from anatomy. And FDA's 2017 approval of pembrolizumab for any patient that has a certain genomic signature, any cancer patient, essentially ushers in a new era in how we can use AI in drug development and really start to focus on the individual patients and the mechanism of disease. So in the 21st century, reclassification of disease involves AI, machine learning, and Nicholas already talked about identification and segmentation, referring to the fact that on this bottom panel you see a pathology specimen that is now being exposed to feature extraction using convolutional neural networks, basically many different layers that extract certain features out of these slides. And the output, in fact, are features that are not visible to the human eye. In fact, now we already have the ability to extract features from pathology slides and start to reclassify diseases, meaning start to identify patient groups, individual patients, and categorize them in a way that is otherwise not possible by just looking at the slide, for example, under the microscope. And that's where a lot of the low-hanging fruits for AI are, and it really speaks to having a more holistic approach to analyzing all the data around the patient and extracting features from that data that would otherwise not be possible in order to identify the unique attributes of each individual patient, so we can continue our progress in developing precision therapies. Thank you.
Thank you, Sean. Joseph, Joe, would you share your screen? Thanks.
Excellent. Okay, everybody. Okay, so I trust that you guys can hear me. And I see the note. Now. Don't shoot. Extend. Yeah, there wasn't an option. I apologize. It will make this work. Okay, everybody. So, my name is Joseph Geraci. It's a pleasure to be here today and present to you, and to talk a little bit more specifically about how we've been dealing with the challenges that Sean and Nicholas have so exquisitely shared. So Sean has been talking about reclassifying disease and the limits of biomedical reduction. I'm going to show you how we've been attacking this. So let's go through some of the rudiments of AI.
Okay. So what I wanted to just quickly go through is there's various ways to distinguish between the various machine learning methods. And what I wanted to do is give a very simple introduction to AI with no math, even though you can see so. So the way to think about these things, there's supervised and unsupervised methods, and there's shallow and deep methods. For example, Sean was talking about that pathology sample, where you get a lot of different perspectives, all these different layers. And I'm going to explain how this happens. So in the shallow method, what you're doing is you're looking for a simple pattern. You can see here you have a bunch of blue dots and a bunch of green dots, and you can see it's very simple. You can split it in two. Now, please press next. And so what happens when you get something complicated like this? So your brains can see that there's a pattern. You can just draw a circle around those red dots. Right, next slide. And what you see here is there's a way. Press next. You remove the animation. Okay. So go back one. So what happens here is there's a way to actually transform space mathematically. You don't have to worry how, so that you get a simple separation. So there's these shallow methods that are able to learn from smaller data, but what it can learn from are kind of these simple patterns. Okay, now next slide. And there's these other methods, like individual trees. Now, clinicians use trees in their mind all the time, right? right? If a patient has these certain criteria, then they fit in this classification, or they probably will be best suited for a certain treatment. So these trees individually are shallow because they just use a simple way of trying to understand the pattern. But if you combine a bunch of shallow methods together, you start getting deep methods. And what do I mean by a deep method? A deep method is one where you actually are able to capture multiple different perspectives about the data, so that the machine can deal with exceptions. Right? So there's many perspectives that the machine puts together, and so that in the future it can capture exceptions and very precise ways of classifying, like what Sean was talking about, N of 1. The problem here is that you need massive data. Another example, next slide, please. And this is where things have gotten very interesting. So next slide. Next. Yeah. Okay. So let me explain what's happening here. Remember all those individual trees you were seeing. If you were able to combine the ability for each of those individual trees to make a decision, you're able to capture a lot of different perspectives. And with a deep neural network, what happens is you have this input layer, which literally is like variables. For example, variables could be if we're talking about classifying vehicles. It could be the length of your car. It could be how many windows the car has, what color it is. So if you have a very long vehicle with a lot of tires and a lot of windows, and maybe if it's yellow, it's probably a bus. And what happens is each of these hidden layers, kind of like a tree, it captures different aspects from the data. And so there's a variety. What's happening is these networks have now blossomed into, there's all kinds of variety, but it's always the same story. What happens is data comes in, and the machine attempts to learn all of these different perspectives about the data, about the patient population. And what you end up with is an ability to make predictions accurately. Next. And the big problem here is that a huge number of patients are needed for this, and it's good for certain situations. Next, please. But the ability to test hypotheses is very difficult, like this, right? So next. So this will excel in large cohorts and like for real world data, and this is emerging, and this is very exciting in order to make predictions. Next. But what we're doing is we're attempting to allow the human to interact with some of these smaller data sets so that we can share in the insights and the learning. Next. And so basically the machine, right? Wouldn't it be amazing if the machine Wouldn't it be amazing if the machine were able to reveal insights that you can interact with? Right. So next. Okay. So let me show you the difference between these types of learning. So this is a mix of shallow and deep methods. So next. Okay. So this is a cutting-edge neural network learning how to cluster non-small cell lung cancer patients. And you can see that the two groups are separated quite well. But imagine a situation like this, next, where the machine actually reveals not just these two subtypes, but all of these other subtypes. And so I'm going to show you an example now with a video. Next, please. Next, please. And next. I'm going to just jump into the video. Yeah. And so what we're going to do here is I'm going to show you a demo where you're actually going to see it live, working with some non-small cell lung cancer data. All right. So this is a platform we developed in order to address these challenges as we're in the industry trying to get pharma companies to do this. So what these are is that we have different apps, and what we're really going to do today is NetraPlay, which is a way to actually have the machine learn what you asked it to. And for it to discover things that you didn't ask it for. And this is the difference between supervised and unsupervised, right? So an example of supervised, I told the machine, look, the blue patients are squamous cell carcinoma of the lung, the red patients are adenocarcinoma of the lung, and what it does is it reduces from thousands and thousands of genes down to these 16 to help explain this phenomenon. And so you can see in this map that the patients are segregated, but you can zoom in here, and you can actually see that these blue patients, the squamous cell carcinoma, actually cluster well together, and you can actually you can see and hover over a patient and see who they are. So what the user's doing here is they're asking the machine, why did you cluster this group of squamous cell carcinoma together? And so let's ask Google, and let's see what Google says. And what you're going to see is that the reason all these people are coming together is because of IRF6, which has been implicated many times. Okay, so it's a nice story. It's able to separate the two groups. And this is supervised learning, meaning I gave it the labels, I asked it to learn something, and then I can compare them. So now I'm going to ask, okay, what genes are driving these two groups, and it explains that. And this is fascinating. I can even go down into the individual level and say, what's really driving the phenomenon here? Okay, so now what we're going to do is you're going to see that a lot of these models are similar, right? The machine produces these different perspectives. Different perspectives are like those different layers of the deep neural network, but we unpack them for you. And what that does is we do stuff like this. Now look at this. This is an unsupervised situation. We didn't ask the machine to do this. It discovered that adenocarcinoma actually splits into these two groups, and there's that other third group up top. But let's ask the machine why this is doing this, and this is fascinating. This is a highly significant variable that comes out, and what it is SLC6A8, as you're going to see in a second. And this, okay, it's been implemented for non-small cell lung cancer, great, but what's amazing here is that it's a differentiator for aggressive and adenocarcinoma. And so the machine's actually able to see these differences, and this has allowed us to really dive in and understand disease like never before. Right, we've been able to unpack certain things in neurodegeneration. We've been able to help pharmaceutical companies and so forth. And this combo, so go back to the slides, please. So what this has allowed us to do is, it's allowed us to intervene with clinical trials to understand what is actually driving the phenomenon. Next. Let's just go through this slide, So what we've been able to do is use this technology to come up with placebo response. Next, please, and next. Yes, here. So what we've been able to do is we can involve when a trial fails, for example, and we utilize phase 2 data, and then what we're able to do is actually interrogate, help the pharmaceutical company interrogate the trial and say, listen, these are the type of patients that are optimal. We've been able to discover placebo response predictions. I'll show you a quick example of that. And then going to Sean's points on adverse events, we were able to get them right down to the individual, right down to the individual for discovering adverse events using technology. Right. So next slide. And of course, real-world time series analytics. So the unique platform, this has allowed us to match drugs with specific subpopulations and even to specific individuals because of the resolution here. And I'm not going to get into how we've been able to do it, but it's basically created a novel method to merge these amazing machine learning technologies to actually deal with smaller numbers and to be able to learn quickly from and have humans extract information. Next, please. So next, down to the bottom. Okay, so this is an example of the power that this type of method brings. So what we did is we fused, and one more, perfect. So we fused data sets with AI, with systems biology. And what this has allowed us to do is it allows the AI to extract subpopulations, explain the phenomenon that's happening at the individual subtype level, and then do systems biology, figure out where drugs can target. And the machine has accurately been able to figure out the rudiments of dementia and actually make predictions about what can go into trials. And so we're using this now in several different ways to come up with new models. Next slide, please. Here's an example of placebo response that we've been able to do. So next, go down to the bottom. Yeah, nice. That's perfect. And so what happens is this is a fascinating case study I wanted to share with people. And what happened here is that we had this bipolar data set, and it was equipped with many different scales. And the machine extracted these items from the scales that boiled down to attitudes, and it was able to accurately predict placebo response, placebo non-response, placebo response. And we're continuing to do this work and start utilizing it in the industry beyond academia. Next slide. And this is an example where I think that we can. Next, please. Where we can actually hone in on what and next one more. What Sean was saying, and this is basically not only did I show you our ability to reclassify disease, but the point that there's no average patient. And one of the critical things that we found is our ability for the machine to understand that certain people belong together. And what happened is we were helping a pharmaceutical company with a clinical trial, and there was an adverse event that occurred. And we were able to do is go back to the machine, pull the patient that they were asking about, and found that there was another patient just like them. And there was these cardiac variables surrounding these two individuals. And this kind of interactive AI that merges, classical machine learning techniques, shallow and deep methods, with this novel mathematical paradigm and this ability to empower clinician scientists to engage with the data. This has allowed us to actually get down to the individual layer and to reclassify disease and to, in pharmaceutical clinical trials, improve the way they think about their drug and how their drugs can match with potentially efficacious populations. Right. So I think I'm done. Next slide. Yeah, that's it. Thank you, everyone, and I'm sorry about the technical difficulties.
Because of the time limitation, we will not take any questions. But here's our email address. Please provide your questions, comments, and feedback for this session to our email address. As Lisbeth mentioned, there will be a sequel for this discussion. This is just a kickoff for our discussion of this topic. So we would like to hear back from you in terms of the topics and questions, what you'd like to hear as we continue discussion on this topic with all of you. Thank you again. I'm passing a virtual microphone to Dimitri for next session.
Hello, everyone. Good morning, good afternoon, good evening. Thank you, me and Nicholas, Joseph, Sean, for the great introduction. We've tried to pull together a list of use cases to follow suit from the introductions into very practical applications. Okay, so we've selected a list of use case presenters to represent the pharmaceutical industry, to represent academia, and to represent some technology vendors in the space. We are hoping that it's a very nice cut across the various areas, including its application in a wide range of spaces, from basic clinical operations execution all the way through into more complex, real-world evidence applications for placebo arms and things of that nature. Our speakers, in the same order as they will be presenting, are Prasanna Rao, who's the head of AI and data sciences for data management at Pfizer, manager in our oncology data management team at Pfizer, Willie Mulhausen, who is a PhD candidate at Dublin City University and a member of the SOUNDS Foundation, which is Ireland's Insight Center for Data Analytics, Emma Richard, who's the manager of Advanced Data Science Insights at Janssen Pharmaceuticals, Ruthie Davey, who's a statistician and vice president of data sciences at Acorn AI, a metadata company, Karen Hoffman, associate principal in the AI and Analytics Intelligence Guild of Cognizant Technology Solutions, and Julian Isla, who's a resource manager for the AI group in Microsoft Consulting Services, and chairman and co-founder of Foundation 29, an NPO focused on how artificial intelligence can empower people to make decisions about their own health based on evidence provided by data. Next slide, please. So this is the same. The sequence is in the same sequence as the presenters I just introduced. I would ask each of the presenters to leave their camera on when they begin their session, and then they can feel free to either leave it on or turn it off while the slides are being shown. Our email representative will forward the slides when you are ready. And if we have time, we're planning to have a 20-minute Q&A session at the end of all of the use cases. And I think a very important factor before we launch is that a critical filtering mechanism when we pick the use cases is that these are all actually implemented. These are not theoreticals. So hopefully we can share some industry-wide experience here. So Ashley and Prasanna, you're up.
Thank you, Dimitris. Can you hear me? So I see the presentation is coming up. So I'll quickly introduce the use case, and Ashley will fill in the details. The use case is what we call as a smart data query. That's a term which you use to talk about how we can optimize data management by looking at discrepant data and being able to identify the discrepancy in the data using machine learning. So let me proceed. The use case begins with the sites entering data into the EDC system, and data managers review data in the EDC system. And today they're using manual techniques, including spreadsheets and other reports to identify the discrepancy and raise a query to the site to confirm or change the data if there's a discrepancy. So what we have done is we have taken that process and we've introduced a human-in-the-loop process with machine training done by data managers. So that is the use case, and I think the presentation is still not being displayed. So as a part of the use case, we have trained the machine over several different iterations and several different domains. An example would be, let's say we were trying to see if there's a logical match between an adverse event and a concomitant medication, right? So let's say there was a drug called loperamide given for treatment of, let's say the adverse event was headache, okay? And the data has been marked as associated with the concomitant medication of I'm seeing the presentations are still changing. We're not yet in the right presentation. So while we pull up the slide, so the use case scenario is matching adverse event with concomitant medication as an example. That's just one scenario. So that requires a lot of data for identifying discrepancies and some clinical inference to understand the relationship between concomitant medication and the adverse event. So that is one of the use case scenarios where we found a combination of semi-supervised learning as well as clinical inference models were working in tandem to flag any discrepant data, and the human in the loop process was looking at that data, and the data manager was able to then conclude if the concomitant medication was a logical match with the adverse event or not. So that's just an example of how we have built the system. And besides these two domains, there are several other domains and several other types of discrepancies the same model can predict, right? So we found that the models to be very accurate. And we talk about accuracy on how we measured it. And that's a classical, confusion matrix approach where we've taken several million data points, and from those data points, any of those historical queries that were raised by the data manager were compared, and we plotted the match between the machine versus the human in a confusion matrix. And we found that the models for the trained domains were in the 85 to 90% accuracy range, right? So that is just the start. And as we provide more and more feedback by utilizing the system in an operational setting, we are able to continuously improve that accuracy of those models. And this is, since this is a human in the loop approach, this is not a continuous machine learning, right? So we do collect all that feedback, and we have expert humans who look at that feedback from different data managers, and that is then used to retrain the models in a very controlled setting as opposed to letting the machines learn by themselves. So those were actually my first 3 slides. Seeing if the presentation is still not being shared. So with that, I will turn it over to Ashley in the interest of time, because we'll talk about how Ashley and his team taught the machine, what we call as a teach cycle. And the teach cycle is the process where data managers train the machines. And so over to you, Ashley.
Thanks, Prasanna. Can I just confirm, Prasanna, can you hear me?
Yes, Ashley, can hear you.
Ashley,
okay,
just one second, please. Ashley,
sure.
Should we just share the slides, because at this point we're halfway through the presentation and the slides are still not up. Let's see if this is them. Otherwise, perhaps we can share.
I have them, Dimitris, if you need me to just share my screen.
Let's see if this. No. Actually, if you're able to, please go ahead and share the screen.
Yeah, let me do that. Can you confirm you can see?
Yes.
All right. Let's hope this stays stable on my screen. Okay, so thank you, Prasanna, for the introduction there. Prasanna just kind of linked into what I wanted to talk about next, which was for our use case here, how did we develop what we call the teach cycle, i.e. the developing that human in the loop, and in this case, a data manager in the loop, in the model's output, and providing the feedback to what the model had developed, what it had predicted, so that it could retrain and of course improve that accuracy. So what we call the teach cycle here was defining the data criteria that Prasanna used, an adverse event and CONMED example there. We are exploring many others. But for an adverse event, cross-checking against a concomitant medication, there's many different checks that are performed within that overall data review as a data manager. We're looking at dates, we're looking at names, we're looking at indications, we're looking at key identifier numbers within the CRF data. And so by breaking that down into what we call subcategories of data review, we were able to provide a definition document that essentially the model would be built from. That plus the next step that you see down here, this ground truth, which is essentially that labeled data, giving the machine examples of historical queries. That's the questions that we raise to our sites when we see a data discrepancy. Examples that are labeled with those subcategory numbers of queries that fit into our expected outputs from the model. The model is developed, the model is refined, and we see data predictions. These are predictions that are data discrepancies. There's something not right with the data that fits into one of those subcategories that we had fed in at the beginning. And not only is the model telling us as data managers that there is a data discrepancy, but also it's providing us that explainable AI. Why is there a data discrepancy? It's giving us the reasons for why the model came out with a prediction. This is the key part of a DM or human in the loop feedback. We have a data manager that would then validate that prediction. Essentially, a thumbs up, thumbs down, very simple approach as to whether it was a good or bad prediction, whether it was a true or false data discrepancy. And linking into that, we can then raise a query to our investigative sites directly from that prediction itself in the user interface that we have designed as a part of this project. So not going to go too much into the details. I know we don't have too much time here, but just a recap of subcategories. When we talk about some of these very complex, these manual data reviews that have been a part of our data management industry for many years now, the reviews form a part of what's a number of different sub-reviews. Like I say, when we're looking at adverse events and comments, there's a lot that makes up that overall review. There's a lot of different data points being looked at. There's a lot of different decisions to be made as a data manager to ultimately decide at the end here, do we want to raise a query, a question to the sites on that data? Once we have provided that subcategory list and we are able to label some examples of our historical data in what we call ground truth, we have data managers that are able to tag that ground truth with the subcategories and provide the model with a starting point to develop its algorithms. Validation, of course, as I just touched on there, is an essentially important piece of what we do to ultimately improve accuracy all of the time. We want to not only make a decision about, as a data manager, whether we should query the site on the data discrepancy that we're seeing, but also we want to provide that all-important feedback to the model. It needs to know, of course, whether it is right or wrong, and where it is wrong if it is wrong. So we've developed a very simple mechanism, trying to keep it simple for the model, but ultimately for the end users, which are the data management team. And we have a toggle, a green-red toggle approach. Was this actually a discrepancy? Yes or no? Was it the right kind of discrepancy? Linking that back into those subcategories we developed at the start of our teach cycle. If it wasn't the right kind of subcategory, then let's tell the model what kind of subcategory that actual data discrepancy that was predicted was. And what we've also built into what we are doing as part of this Smart Data Query project is one of the most manual pieces of a data manager's review of these data types is creating the query text, the questions that we want to ask the sites. The way it is done historically is we review the data manually, we write a manual query, and we manually raise that query on the data point we want in our EDC databases. What we have combined here with SDQ is the use of natural language processing to actually develop an intelligent query text that we attach to the data discrepancy prediction that the model makes. And so if the data manager likes the query text, thinks that that would be meaningful for our sites to be able to address the issue, they are able to move to the second piece, which is a simple thumbs up, thumbs down, or on hold approach. Thumbs up being this is a good prediction, it's definitely a data discrepancy and we should raise a query to our sites. Thumbs down being that there is a reason we would not want to raise that query. Maybe a CRA was performing a monitoring visit just this morning and has already raised a query. Maybe we don't want to duplicate that querying effort. But for some reason, we don't want to raise a query on the data discrepancy prediction that was made. Or on hold. Maybe we need to take some further action before deciding on that approve or reject decision. Maybe we need to follow up with our medical leader. Maybe we need to follow up with our clinician before we do before we take that affirmative action of approve or reject. So there are a number of different operational considerations for the implementation of SDQ, or Smart Data Query as we are terming it. Of course, we need data, and we need labeled data, and we need volume of labeled data. So ground truth generation and taking our historical discrepancy data from historical studies to label that with those subcategories that we had defined in each teach cycle is key to being able to develop those models and develop the algorithms that are needed. We need to, of course, have a robust and concrete teach cycles, the pieces that we had just shown there on a previous slide, to ensure that we have the right machine training being developed using the labeled data, to ensure that we're able to run those models and evaluate those models. It's key not only to provide a data manager with a data discrepancy prediction, but also to have that explainability as well. That's all important for a data manager to be able to make that decision as to whether this is a good data discrepancy prediction or not, and whether we would want to raise that query to the sites or not. And then we need to embed all of this into our operations. This is, of course, a very novel approach to how we clean very complex and traditionally manual data scenarios. So of course, scaling up our models to ensure that they are fit for the data of our 21st century trials, to ensure that the process of utilizing Smart Data Query is embedded into our processes and into our existing SOPs and control documents, to ensuring that we're able to keep a track of how the AI is performing and to be able to evaluate the benefits from it. And most importantly, from that human in the loop feedback or data manager in the loop feedback, as we're considering here, to ensure that we've got that continuous retraining, that continuous learning, so that we can reuse those models across trials in different therapeutic areas and indications. Thank you very much, everyone.
Thank you, Ashley and Prasanna. I think up next is Willy Mulhausen. If the slides are ready to be shared from Emma's side, or do we need Willy to share the slides directly?
I will wait. I can probably just get started. So, my name is Willy Mulhausen. I'm a PhD student at Dublin City University, and my presentation today is about signal detection and wearable device data. I'm part of a working group around Professor Timos Ward at TCU, together with my colleagues Owen Brophy and Lily Zhang. Today I want to talk about wearable devices and how and why we looked into wearable devices. As most of you will have noticed and have seen, that wearable devices are everywhere these days. Number of projects across clinical research and healthcare is growing. There's even a lot of interactions right now to turn wearable device data into primary endpoints for clinical research. There are groups out there that support the development of new digital endpoints. there's user groups within ISPOR, IsoQual, DA, and others, and even dedicated new groups like the Dyne Society. So wearables are basically everywhere, and you can't get away from them to a certain degree. One of the challenges I've seen in many years that I've been involved in wearable devices, and I've done my first project in 2005 with wearable device, is the ALCOA acronym, which stands for Data Has to Be Attributable, Legible, Contemporaneous, Original, and Accurate. I've been involved in clinical research since 1998, and that was one of the very first acronyms that was basically hammered into my head at the time. And this attributable is, in the context of wearable devices, always an interesting one. So basically, we have to make sure when we capture data that we can say who actually entered the data, who moved the data, changed the data, and so on. And most of you will be very familiar with that. So with wearable devices, with our current data capture systems, whether it's an EDC, ECOA, clinical systems, or IVR, RTSM, and so on, we usually have some form of login where users have to use a username or a combination of username and password or PIN. And we all do that multiple times a day as we're involved in these things. So that's practiced by now. But how do we do that in wearable devices? We can see that in wearable devices, there are quite often there's a combination of an app or a desktop application and a device. And these devices are getting smaller and smaller and then getting bigger again, but usually they're very small. And they don't really have a screen that we can interact with. So login on a device is very difficult. So most of the time you will have to set up an account and log in when you set them up, usually on the app on your phone. And then probably sometimes you have to redo that when the session starts, so when you start recording data. But then what happens when you're in the wild, when the patients are actually out at home? Who is then going to control that they're actually doing it? So the real question is, who's actually wearing the wearable device when patients are at home and trying to capture data and submitting data? So one of the first projects, well, there are my slides. You can go now. The next one, please. And I think that's where I am. One more. And another, and another. Here we are. Thank you very much. So this here, nope, go one back, please. This image was dark and a wearable on its neck there. That's where it all many years ago. Rumor had it in multiple conferences at the time that patients would basically generate data by putting the devices on their leash, for example, or on any kind of hardware. Me being a veterinary researcher, I decided that's something that I should look into. So with my colleagues at the time, we basically gathered 3 dogs, the owners, 4 dogs actually, 4 owners, and established or equipped everyone with a device. So the owners, so the humans and the dogs all got their own device, and then they went off for a walk and generated. And what we found is that actually dogs could generate data on how tall the dog was, so in the stride length, we would see better quality data, or not so good quality data. But what we also found is that we could clearly separate, when we look at the raw data, we could separate the humans and the dogs. So we published that, but that wasn't signed enough. Some journals so decided not to take the publication. But what we also saw in the same data set is actually the different humans from each other. And that was actually very interesting. See that people can't hear me properly. Can you hear me now? Is that okay, or is it getting better? I have mine actually on as well. I don't know if you can hear me now or not. I'll just try to speak a little louder, so probably that is okay then. So what we did, we found that we can separate the humans, and based on that, we tried to do some more research, and I'll get to that in a second. So when you do look on the internet, you will find a lot of hacks, basically, in articles on how people try to cheat the system by generating more steps in their film. There were systems out there, like on the bottom left screen, you can see that that you could buy a device. You put your mobile phone in there, and it would basically generate data on the mobile phone, pretending that's on a continuous basis. On the right-hand side, you link to a YouTube video where there are instructions on 4 or 5 different ways how to generate data or wearable device data to generate data. So there seems to be quite a bit of activity in that space, and therefore we have a problem, allegedly, put it that way. Next slide, please. So in a long time ago, there was an initiative where Inception was developed, which is a convolutional neural network, and it was developed to assist in image analysis and specifically in object detection. So the challenge was that the system should be able to differentiate dog images or images of dogs and images of a cat. So the system was smart enough by analyzing images to say whether that was a dog or a cat. Next slide, please. So what we figured, if a system can do that, then why don't we try something similar? So when you've seen the raw data from wearable devices, you would see quite often you get these tables or with a lot of numbers in them, depending on how much data density you collect. You'd be millions of rows. And we found ways, there are ways to turn these numbers into pictures, basically, into images. So that's what we did. And here on the bottom, you see the time. So these are 3 seconds of 3 dimensions of the actigraph device. That's basically accelerometer data. Next slide, please. So we then moved the noise, basically the frames and all the other stuff that needs to go, to just basically look at the image. Next slide, please. And what we then did is we had a hypothesis that the numbers, so as you turn the numbers into images, you keep the information, and it's a classification problem, which means we want to know that these 3-second intervals of that is that specific 3-second interval from patient A, B, C, or D. And we have data from 6 patients initially. And we use the machine learning algorithms to analyze the raw actigraphy data that we turn into these. I'm hoping that we could build, or that we would see almost like a biometric fingerprint or an identifier, and use that identifier to analyze data from different and bucket A, B, C, D, or 1 to 6, and see how many we put in the right bucket. So the 3-second snippets that we identify correctly, assigning it to a specific. And then later on, we wanted to check what kind of parameters drive the fingerprint recognition rate. So what can we do to the actual images and the conversion images to make this process better? And we wanted to use multiple data sources, so different device types and patient types and so on. Next slide, please. So what we did is, and what we found is that it actually works. So a couple of posters and one publication in a journal. So the first one, we found that when we use 3D, so 3-dimensional accelerometer data, and do these 3-second snippets, that we could basically 97.9% accuracy turn these or class and basically say they volunteer or patient 1, 2, 3, and 4. So on the you see only 4 that what we did that. After that, we thought, okay, why don't we use this other data? So we use photoplethysmography data, which is inherently very noisy data. That's data that's being generated by your wearable device when you have these 3 green lights underneath the thing that basically shine into your skin and determine what your heart rate is. So this is very noisy data, and we tried that with that data, and that actually worked pretty well too. So the accuracy was not as good enough that we were very happy that it actually, we think it works. And with more work, we can probably get even better with that. We also did it with ECG, and that worked fine too. Again, it was a little bit noisy data. We used a chest belt. That worked surprisingly well, to be honest. But the downside of the 3D accelerometer data, for example, was that it was done with 256 hertz. So it's a very high hertz rate, which is out of data. And this is in practice, in a clinical trial or in patient care, probably not really a good option. So we decided that we needed to look into how can we do that. Can we actually massage the data and the numbers and the images and make it, the sample size smaller, so it can make the data set smaller. So we went from 256 hertz all the way down to 1 hertz. And on the right-hand side of one, you will see that the accuracy went down. But the interesting thing was that even at 10 hertz, we had an 83% accuracy. And again, this is just out of the box, we tweaking or anything. So we believe that we can increase that. And in many cases, 83 is actually a really good result to start with, and maybe good enough. But it's only if not the data that we need for that compared to the 256 hertz. So we will continue to work along these lines and try some more tests. We've got some more on there, but very promising results that we found there. Next slide, please. So next things that we're going to look into is what different parameters do we need to tweak and can we work with when we paint the picture. So when we turn numbers into graphs, basically. So the epoch length, as I said before, we work with 3 seconds. We work now with 6 seconds. We work with overlap. So what if we overlap these epochs? Frequencies, we went from 256 all the way down to 10 hertz. We've done that already. And then we want to combine different data sources, so actigraphy and PPG data and ECG data potentially, see if that increases our accuracy. And then we want to get other stuff like data change over time. So for example, if you do some gait analysis, and if the gait changes when the drug or the procedure works. So what if some of these that changes over time as the patient state changes? The algorithm still detect that or not? So these are things that we're looking into. And we want to analyze more data sets. So we're looking for data donors. So if anyone has some data sets out there that we could use to do some more research, we would highly appreciate that. And if you just contact us. We focus so far on fraud detection, but we're looking at some of their capabilities as well, because the principles would be very similar. So it's basically all about detection of patterns. And we participated in a Parkinson's disease model in a challenge by the Michael J. Fox Foundation, and we did reasonably well. We're still looking at we could find similar results that we found on the fraud detection, so that was very encouraging. And we're now looking at several other therapeutic areas where we can apply the same technologies to basically detect patterns that would indicate a certain state. Could be like cough recorders, one of those we're looking at fall detection. There's a couple of things that we can do in this space, we believe. So thank you very much.
Thank you, Willie. Interesting overview.
Yeah, thanks.
Although we always hypothesize if people put these things on their pets when we're measuring their movements. I believe Emma is up next.
All right. Hello, everyone. I'm excited to be presenting to you today about the ability to enhance protocol deviation trending in clinical operations using natural language processing. Next slide. As was mentioned, my name is Emma Richard, and I'm a manager of Advanced Data Insights in the Global Development Group at Janssen Pharmaceuticals. Next slide. Before we begin, just one note that this presentation is intended for informational purposes only, and statements of fact are not the opinion or position of Janssen Research and Development. Next slide. Today I'm going to review at a high level the executive summary and background of the Protocol Deviation Trending Project. I'll review the process of turning unstructured data into insights, as well as the learning architecture applied to classify protocol deviations. Finally, I'll review the results of applying natural language processing to protocol deviations and how the classifications are currently utilized. And if there's time at the very end, I'll take any questions that you might have. Next slide. First, an overview of the Protocol Deviation Classification Project. The trending of protocol deviations is key to ensure high data quality and patient safety. When a high percentage of protocol deviations are classified as other or unclassified, it's difficult to properly conduct a trending analysis because most of the key information is stored in a free text field, which takes time to manually review. The application of natural language processing techniques to classify protocol deviations into a set group of categories helps to improve study risk oversight. I'll describe in more detail on the following slides how the natural language processing techniques work and how the tool has been operationalized within our organization. Next slide. Again, protocol deviation trending analysis is key to determining the health of the study. Previous processes in place at Janssen R&D made trending analysis a highly cumbersome and manual process due to the fact that key information, again, is stored in a free text field. And approximately 80% of protocol deviations have been classified as other. Working with a small team of central monitoring managers who oversee risk at Janssen, I determined 25 key subcategories and 11 key categories to be used for protocol deviation classification. Next slide. So looking at how we turn unstructured data into insights, the first step that the team needed to accomplish prior to the application of natural language processing techniques was to have subject matter experts manually classify a set of protocol deviations into those 25 determined subcategories. The protocol deviation descriptions can be thought of as the independent variable in the context of this problem, and the subcategory can be considered the dependent variable. Once the data was cleaned, a numerical vector was created using natural language processing techniques to represent the description field, thereby becoming the true X variable or independent variable. So not the words themselves, but the numerical representation of the words in the description of the protocol deviation. The data was then split into trained test and validation data sets in order to develop and evaluate the model. And the training data set was made up of those manually classified deviations. Ultimately, we resulted in a classification accuracy of over 80%, which was deemed acceptable because that's about the accuracy of human beings classifying protocol deviations. You would still expect some discrepancies there. Next slide, please. The first natural language processing technique that was used to create a numerical representation of the protocol deviation was term frequency inverse document frequency, or TF-IDF. TF-IDF is calculated by looking at the number of times a certain word appears within a given protocol deviation and then across the entire set of protocol deviations. Essentially, it scores the uniqueness of individual words. A support vector machine is then used to classify the vector set, ultimately separating the protocol deviations into subcategories based on the numerical representation of that description. So if we look at the plot on the slide, the protocol deviations that have the most similar descriptions would be plotted close to one another, except in multiple dimensions, not just the 2 dimensions shown here. And the classifier is determined as the line when we look at the plot, or in the case of multiple dimensions, the hyperplane that most separates the protocol deviations into their respective categories. You can imagine with protocol deviations that it's not quite as clear as the black and white data points represented on this plot, and it's likely that we would have some misclassification, especially between very similar categories and subcategories. But the same context of a support vector machine is applied when you're separating 25 categories as it is if you're looking to separate data into 2. Next slide, please. Word2Vec was also explored as a natural language processing technique to convert the protocol deviation descriptions into a numerical vector. Word2Vec analyzes the structure and placement of words in context of an entire document or description, more so than it looks at the importance of individual words within a given description or description set. You can see the simplified example here that height and weight have very similar vectors compared to the word drug, because the context in which those words would fall would likely be very similar and followed by words like measure or sample, and drug would probably fall in a deviation with a much different context. Next slide, please. Comparing the two methods of TF-IDF and Word2Vec, TF-IDF produced a test accuracy of 84%, while Word2Vec had a testing accuracy of 64%. And this is likely due to the importance of individual unique words in determining protocol deviations that belong in one subcategory versus another. So, for example, if we compare the two made-up protocol deviations on this slide, TF-IDF would be more likely to correctly classify both of these protocol deviations. So the PDs we have here are the patient did not return the study drug to the site, and the patient did not return the questionnaire at the visit. So TF-IDF would analyze and apply more weight to the phrase study drug and the word questionnaire due to the frequency of those words in their respective category and the uniqueness of them to those categories compared to the entire protocol deviation set. Whereas Word2Vec would be looking more at the context, and here we can see that the patient is a similar phrase that of course would be similar to a lot of protocol deviations, and then at the site versus at the visit. Likely the model would understand these protocol deviations to be related to something around patient compliance. So we would expect that probably one of the two of these would be classified correctly and the other would be classified in the same category of patient compliance. Next slide, please. The confusion matrix for protocol deviation classification using TF-IDF is shown here. And for ease of viewing, the data that I've included here is the 11 categories that the 25 subcategories are rolled up into. The misclassifications of protocol deviations can be somewhat well understood because similar categories are most frequently, excuse me, sorry about that. Similar categories are most frequently misidentified as one another. For example, if we look at IP compliance and IP dispensation, we can see that around 75% of the deviations are classified correctly, and about 7 to 8% are classified into the opposing category. And as you can imagine, when we look down at the subcategory level, most of the misclassification occurs within a single category. So IP dispensation, for example, has 5 subcategories labeled 6A through 6B, ranging from IP not being stored correctly to IP not being dispensed correctly, and most often these subcategories are misclassified as one another. Next slide, please. Once the model was developed, it was deployed into a visualization that study teams now use for protocol deviation trending. So protocol deviations can be viewed by country, by site, by therapeutic area, and also our central monitoring managers are able to view the trends in protocol deviations and their categories over time. Next slide, please. This tool is most helpful to end users because it removes the noise that the other category creates. So you can see here on the left, the mustard color represents the protocol deviations that are classified as other. And on the right, the protocol deviations are more accurately classified through our TF-IDF program so that more action can be taken from the analysis of this data. So this tool really aids our central monitoring managers and study teams and makes protocol deviation trending less cumbersome. And our end users are trained and aware that the predicted classifications are about 80% accurate, so manual oversight is still needed when using this tool. Next slide, please. So in summary, the creation of a natural language processing tool for protocol deviation classification is key to provide actionable insights to study teams. Comparing the TF-IDF and Word2Vec methodologies, the importance of individual unique words that's captured by TF-IDF results in a higher performance when classifying protocol deviations. So as we continue to develop this program, we'll need to ensure that our training set continues to grow with those manually classified protocol deviations, and we will evaluate more advanced natural language processing techniques to ensure that we're continually advancing our model so that our protocol deviation classification and trending can be as accurate as possible and as easy for our end users to complete as possible. And I believe that's my last slide. Great. And if there are any questions, if we have time at the end.
Yep. Thank you very much, Emma. And thank you for being very punctual with your timing. And I believe Ruthie David is up next.
Hey, everyone. My name is Ruthie Davey. I'm not sure should I share my own slides or?
Is this is what we're seeing right now? Yes, perfect.
This is perfect. Okay, thank you. So I'm a statistician. I lead the synthetic control arms group in data science at Acorn AI. And what I have to share with you today is some thoughts about estimating a counterfactual and addressing some of the points that were actually already raised, the fact that we're really interested in an individual and not in a mean or a median or an average response. So next slide, please. So when we're faced with estimating the efficacy of a new medical product, we would ideally know the result if the patient receives that investigational product and the result for that patient if they don't receive it. And then the difference between those two things would tell us how well that new medical product is working. But of course, you can never, almost never, observe both of these for an individual patient. And so what we do is the chosen path is, of course, the factual result that we can observe. The unchosen path, whether they receive the investigational product or not, that is the counterfactual. And so what we do instead is we try to estimate this counterfactual by looking at a comparison of groups of patients. And we assume that the average response of the alternate group is a reasonable estimation of what would have happened to the patients in the investigational group if we had been able to not treat them and observe that result as well. And the gold standard method for doing this has become, or at least for medical product research, has become a randomized controlled trial. And one of the key reasons for this is because the randomization balances the baseline condition of the patients in the two groups, so that the difference between the two groups can be assumed to be an estimate of the treatment effect. Next slide, please. So let's consider this illustration of a Kaplan-Meier curve showing the occurrence of time to death, so the rate and timing of deaths in patients who have been investigationally treated. We can see, when and how many deaths have occurred in this group. And still, with all of this information, it's really pretty unclear whether the investigational product has had a benefit for these patients or not, because we don't know what would have happened had they not been treated with the investigational product. Next slide. If you add, though, the estimate of the counterfactual, and the common way to do this is with a randomized control, we can then clearly see that the investigational product has had a beneficial effect. Next slide. So in summary then, I want to point out that the need is to estimate the counterfactual, and the gold standard method for doing that has become the randomized controlled trial. But a randomization is not always possible. And so I want to suggest today that we can address this need in certain areas without randomization. So there are cases where the rarity of the disease make it very problematic for maintaining a concurrent control arm. There's also cases where the ability to investigate an agent is compromised by the availability of that agent or a very similar agent in the same drug class outside the study. And I've listed 2 examples here. There's many of these, especially in the investigation of oncology products. The BRAVO study in BRCA-positive breast cancer was a study of a new PARP inhibitor, was randomized controlled trial, and was actually terminated early when the approval of another PARP inhibitor occurred, and a very high number of the control arm patients left that trial to receive the PARP inhibitor on the market. Sunitinib malate is an illustration, again, a randomized controlled trial looking initially at progression-free survival, and a very large effect was demonstrated. After that analysis, though, 84% of the placebo patients elected to receive sunitinib malate, and not surprisingly, the evaluation of overall survival was not meaningful, did not reveal a treatment effect. And so we want to suggest that some other approach is needed for estimating the counterfactual. We think one approach here is a synthetic control arm. One way to define this, which is how we currently define it, is direct use of patient-level data from multiple previous clinical trials in the same indication. So this would be patients who meet the same eligibility criteria as the eligibility criteria used to recruit the investigational patients. And importantly, we will use propensity score methods to assure that the baseline composition of that group is comparable to the baseline composition of the investigational arm. But this creates a key challenge as well, because we have now very precisely defined the type of historical patients that we need to identify in order to create a synthetic control arm, and leads us to the possibility that more sophisticated predictive modeling using a wide set of patients with some more variety in their baseline condition and eligibility criteria, that we could create predictive models that could predict an individual subject's response, and those predictions could be used in a synthetic control arm. For now, though, I'm going to start with the description of a synthetic control arm using the observed patient-level data. Next slide. So imagine, as we did in the beginning of this presentation, that we have a set of investigationally treated patients that is uncontrolled. There's been no randomized control.
Into an automated voice messaging system. 24068718807 is not available. The mailbox is full and cannot accept any messages at the
Okay, and so we'd like to create a synthetic control arm for this data so that we can better understand the impact of the investigational product. Next slide. We're going to build this from historical clinical trials data. In this picture, the trials are represented by the large circles and patients are represented by dots. So the first step is data standardization across trials. These trials are collected in all different formats. And so we have to remove those boundaries in order to look at individual patients. Next slide. So we do that data standardization. Now we have a set of patients where some of them meet the eligibility criteria and some of them don't. In this picture, the yellow patients are the ones that don't, and so we remove those. Next slide. So now the blue patients and the green patients have all met the same eligibility criteria, and we balance their baseline characteristics like age or disease severity using propensity score matching or weighting. And so in essence, we are choosing the right historical patients to balance that baseline composition. So for example, if there's a male who's 65 years old with an ECOG score of 1 in the investigational arm, then we're looking for a male with a 65 years old with an ECOG score of 1 in the historical set. This is a bit of an exaggeration because, of course, we're not using the individual characteristics like this. We're using a model to estimate these overall. Next slide. So once we've selected the right patients, now we have two very comparable sets of data with balance in the baseline characteristics. The green patients have been treated with the investigational arm and investigational product, and the blue patients have not. Next slide. So once again, here's the Kaplan-Meier curve for those investigationally treated patients. Next slide. And here's the result for the synthetic control arm that we created out of the historical patients. Now we see a very clear benefit of that investigational product. It gives the context that we need to understand that. But to really believe that this is the right treatment effect, that we've made the right estimation, we have to believe that the synthetic control arm is approximating a randomized control. Next slide. So we have to believe this, that these curves would essentially replicate, that the synthetic control arm would replicate the results of the randomized control. And so I have two case studies to share with you where we looked at this question of whether a whether a synthetic control arm can replicate the results of a randomized control. Next slide. In these case studies, we built the synthetic control arm as I've described, with the exception that we matched it to the randomized control. Next slide. So we're matching the historical patients to the baseline composition of the randomized control, and then we're comparing between the synthetic control arm and the randomized control. Next slide. This is the result we would like to see from these case studies, as the synthetic control and the randomized control line up well. Next slide. The first case study we did was in non-small cell lung cancer and utilizes historical clinical trials data. We defined one trial as the target trial and then used all other available trials to build the synthetic control arm. We selected patients who met the key eligibility criteria, and we required the appropriate study treatment to address the research question. In this case, it meant assignment to docetaxel. So on the left, you're seeing the Kaplan-Meier curves before we've done our propensity score matching to build the synthetic control arm. In red, you'll see the randomized control from our target trial. So this is what we're trying to match to. And in blue, you'll see the result for all available historical patients who met the eligibility criteria. So from this picture, you see that without balancing the baseline composition of the two groups, there's a bit of a mismatch in overall survival. There's separation of the curves. Next slide. But if we apply the appropriate propensity score approach, we then match the result very, very well. The curves overlap. The synthetic control arm and the randomized control overlap. The log rank test is insignificant, and the hazard ratio is estimated to be at approximately 1. Next slide. We did a very similar experiment in multiple myeloma. We defined one historical clinical trial as the target. In this case, so one trial as the target, and then all other trials were used to build the synthetic control arm. In this case, though, we're also able to examine the investigational arm to look at the actual treatment effect, unlike the last case where we were focused solely on the control arm. So I'll orient you to this graph. Again, it's a Kaplan-Meier curve for overall survival. And you can see the estimate of this was a successful trial. You can see the estimate of the treatment effect here by comparing the blue arm, which is the randomized control, to the light red, which was the investigational arm from the historical trial. So that's a statistically significant result and was a successful study. The first thing to notice is that the green arm, the synthetic control arm, overlaps very closely again with the randomized control, suggesting that in a future trial you could use a synthetic control instead of a randomized control and get a very similar result. And then the second thing to notice is that the treatment effect is essentially unchanged when you utilize the synthetic control arm versus the randomized control. You can see this in the picture. You can also see this in the inferential statistics, where in the original trial, the hazard ratio was estimated to be 0.74, and in the analysis with the synthetic control arm, it's estimated to be 0.76. So both of these case studies support the possibility that a synthetic control arm could be used to augment or replace a randomized control, especially in cases where randomization is ethically or feasibly challenging. Next slide. So in summary then, I think I hope what I've shared with you is that, the need is to estimate the counterfactual. This is a necessary part of understanding the effect of new medical products. And ideally, we would be able to understand it on an individual patient level. That point was made a couple of times by other presenters as well. Randomization is the method that has become the gold standard to do this, but we think there are cases where randomization is quite problematic, and there are other methods that can be considered here. And of course, AI and predictive modeling are a key part of this. And what we've shown so far is that a synthetic control arm built from patient-level data from historical clinical trials with using careful procedures for balancing the baseline composition can estimate the randomized control. We've shown this in 2 case studies, one in non-small cell lung cancer and one in multiple myeloma. And this provides an opportunity to use these synthetic control arms in future trials without compromising the understanding of the medical product. And then finally, we see great opportunity to extend these methods to AI-based estimates. This would solve one of our key challenges for the current approach, which is the key challenge being that identifying the historical data in such a precisely defined set of patients can be difficult, but the predictive modeling and AI approach may help to widen that net. So thank you very much. I'll be happy to take questions at the end.
Thank you very much. I believe Karen Hoffman is up next. Karen, if you want to turn on your webcam and take over the session, you have the next 10 minutes.
Thank you, everyone. I'm Karen Hoffman. Today I'll be talking about using machine learning to help streamline the complaint handling process. So if we could go to the next slide, I'll start with an overview of what we were doing here. So this was work we did in partnership with one of our medical devices clients. They were looking to streamline and minimize manual work in the complaint handling process. And they were also helping to improve product experience coding accuracy. So for those of you that don't know, the complaint handling process is incredibly manually intensive. Complaints come in, the complaint is described in a free text format, and based on that, folks need to read through the complaints and categorize them in a couple different ways. The first is around the product experience, so what actually happened with the product. Did it break? Did it bend? Did something catch on fire? There's 1,000 different things that could go wrong with a medical device. So it needs to be classified accordingly. And the goal of this project was to attempt to automate that so that they didn't all need to be manually read and coded manually. And just to give you a sense of the magnitude of the problem, the client we were working with deals with 19,000 complaints per month. That's how many are coming in on average. They have over 16,000 distinct products in their portfolio, so a lot of different products are in the mix. And in terms of the volume of product experience codes that could be applicable, there were over 3,000 in their system. So it's a very large problem that we were dealing with. So the approach we used to address this business problem was a couple of things. We started with natural language processing to take the free text event description data and convert it into a more structured format that could then be fed into various machine learning algorithms so that we could train models to help with the prediction of the product experience codes that are applicable to each complaint. And then the other thing we also needed to do was operationalize this. So after we built the models, it needed to be fully integrated with the client's existing enterprise complaint management system so that as the humans were going about their process workflow, they could be presented with what codes could potentially be applicable and apply them to the complaint. So that's what we were looking to do in this project. If we go to the next slide, please. So real quickly, again, the overall process flow is very manually intensive. There are a lot of hands involved in the process. So the complaint comes in from the customer, either the patient or a caregiver or a physician. We'll submit the details of what happened, when it happened, what product was involved or products were involved. An intake user will receive all the information and get it entered into the system as a new complaint. And then from there, a super user will go through and manually read through everything and do that categorization. The reason these categorizations are so important is because it's what ultimately then determines if something is needs to be reported to the EMA or the FDA. So does it need to go for regulatory oversight? So once the super user has made those decisions and entered it in, it goes to the regulatory user who can review, submit reports as and when needed. There's also a product investigation user involved. If the product was returned, there will be engineers involved to analyze, what specifically went wrong with the product, how did it malfunction, and throughout all of this, additional information may be entered into the system. So as more information is uncovered, notes are added into the complaint, which could also then change the coding that's already been done. So that's another issue they're dealing with as well is, how do I make sure that at the end of the day before I close the complaint that we're closing it with the most relevant codes that are applicable. A regulatory reporting user needs to get involved again just before closure to make sure that if anything has changed, all of the reporting is still accurate. And then finally, the super user will wrap everything up and close it out. So that's the overall process for complaint handling. On the next slide, I'll go through what the process was we followed in our project. So it all started with the data collection. Luckily, in this instance, we did have good, rich data to use for our modeling because of the volume of products and complaints that our client is dealing with. So they were able to provide us with 2 years of historical data across all of their different operating companies. We took that data, we went through different cleaning processes and started to apply different natural language processing techniques, removing stop words, applying lemmatization, and other approaches just to try to clean and structure the data in a way that would best then feed into the machine learning algorithms. From a modeling perspective, we started with a random forest approach and quickly realized that there were limitations that were going to prevent that from being successful. We were running into memory issues, and the volume of categories that we had or codes that we had was just too much for random forest to handle. So we then quickly switched gears to a deep learning sequential model using TensorFlow Keras, and this was able to suit our needs nicely. We did various model iterations on different ways to slice and dice things, and ultimately ended up with a pretty accurate model that we felt comfortable with. It ranged across operating companies. On the low end, we were at about 86% accuracy. For one of the opcos, we were able to get all the way up to 98%, just due to the nature of their data. On average, we were around about a 92% accuracy in terms of predicting the applicable product experience code. So we were pretty happy and comfortable with where we landed, although we are building in a quarterly refresh process as well. So as new data comes in on a quarterly basis, we will go back and retrain the models to make sure that they are staying up to date and current with the latest codes and products as new information gets added into the system. In parallel to all the modeling activity, we were also building out the UI so that the users could get the recommendations and predictions from the model and then apply them. And then finally was the integration and rollout. So if we go to the next slide, I can just give you a quick glimpse into what this actually looks like for the user. So the next slide, please. So this is what the user is now going to see in the system. There's a new section that's been added called Code Prediction, where they can click on the Generate Prediction button. And when they click on that, what's going to happen is it passes all of the pertinent information from this complaint to the model. So the product family, the event description being the most important, will all get sent in a JSON file to the code. So if you go to the next slide then, please. So now that that's been passed to the model will return back on the screen what are the 3 codes that are most likely to be applicable to this specific product within this complaint. So you can see it's going to show the level 2 and level 1 codes, and the user then has the option to select or not select the codes that they want to apply to the complaint. So after they've decided what codes they want to apply, they'll click on the Add Selected Codes button. And if we go to the next slide, you'll be able to see that now under Product Experience Code, you can see that those codes that were selected above have in fact been added to the complaint, and they can save them. The user also has the ability, if they're so inclined, to continue to add codes manually. So if there's something that's not showing up in the prediction that they feel is also applicable, they can go ahead and add it. And like I said before, because we have that quarterly process in place, we're going to learn as we go, and the model will get even more accurate over time. So if we go to the next slide, I just want to give you a sense of what else is to come. So
hi, this is the personal mailbox of Dr. Joseph Geraci from Netramark. Please feel free to leave any business or personal messages here. Thank you.
Okay. So this is the work that we've done to date. There is more work that we're looking to partner with our client on in 2021. One of those things is leveraging optical character recognition for any attachments that may be tagged to a complaint. So sometimes I may not type everything into the system as part of the event description. I may attach documents or pictures or something like that. So they're looking to have us leverage OCR to review those attachments so that we can then convert that into structured data and help to aid in that product experience code prediction. They're also thinking about making this a little bit more automatic. So right now, as you saw, there is still very much a human in the loop component to this. Nothing will get automatically assigned as of yet. But if we are able to achieve a high enough prediction accuracy on selected codes, they are considering auto-coding those to complaints rather than the human having to assign them. They're also looking to consider having us work on models to predict complaint closure, because adhering to SLAs is really critical, especially if something is reportable to one of the regulatory bodies. So if we can predict complaint closure and kind of give them a heads up on complaints that are likely to be problematic of achieving the SLA, they could give them special attention. And then the final thing is to potentially create complaints from pictures. So if an image comes in, we could process that, match it to an issue, and prompt a series of questions for complaint creation. So these are some of the things we're looking to do in the future, and that's all I have. Thank you for your time.
Thank you very much, Karen. I believe next up is Julian. Julian, if you're on, please feel free to turn on your webcam if you'd like and activate your microphone.
Thank you for the invitation. My name is Julian Isla. I work for Microsoft in Microsoft rare disease community in Europe. From a regulatory point of view, just to let you know that I'm a member of the Orphan Drug Committee and Medicine. This is my declaration of interest and my so try to be with some examples of artificial intelligence, explain some use of artificial intelligence in regulatory science and the challenges that we are facing using artificial intelligence. Right here, right now, the most, because if you are using, Google Assistant or Siri or any kind of assistant, you're really using artificial intelligence. If you are watching movies on your TV, you probably are using artificial intelligence even if you don't know. For recommendations are based on machine learning algorithm, and the most scary thing is managed by. The question is the workflow? There is voicemail over. I don't know if you guys can.
Let's hope that was the end of it, Julian. Sorry. I think we're okay now, Julian, if you want to continue.
At risk, probably it is. Most of the jobs right now could be on risk in the future because machine learning can really take a lot of works that we are doing right now by humans, and in the future, science are, for example, how deep learning, machine learning algorithms are finding new drugs, especially the big advance of this, because right now we are able to model how the proteins are folding, which is a very, very important scientific problem. And right now the new algorithms, they are able to do this work in a very efficient way. And probably this will change the way the drugs are being. And this will raise a regulatory science issue because at EMA we will need more experts in order to understand how these drugs are being developed and the plausibility of the clinical efficacy. The industry is also changing because of that, because on top of the traditional pharma corporations, there are new players like the AI companies. For AI and providing expertise where the traditional pharma companies or the BD expertise. So probably the landscape of companies will change next years. For clinical trials, okay, the previous speakers already saying, text analytics, which is a product, commercial product from Microsoft, is used to understand medical tests and extract knowledge. This is a medical record. It's a real medical record. It's in Spanish. I'm just copying some text for this medical record. I'm doing a very basic translation into English, using one commercial translation software, nothing sophisticated. And I'm putting the text over. And if I click on eyes, right here, the services analyzing the test, identifying organs, identifying examination, the values for the examinations in a very, very fast way and very, very great. this is a new technology that appeared just this summer. It's totally new. But the thing is, the view is super fast. Every 2-3 months, we have new products, new features that they are able to do that 1 year ago was impossible. Machine learning can be also used for anomaly detection, because it's very important problem. But this can be used, for example, for clinical trials. If one of the data that you are taking from the clinical strains, machine learning algorithms. And during regulatory process, most of the knowledge, most of the work that we are doing is based on documents, in Word documents. The problem is if you are using Word documents, the Word documents like having medical records with no structure. Where is the knowledge? The humans, they have to read all the document, and extract this knowledge. And this is done over and over. The industry is moving into a direction because right most of the knowledge in science is annotated in something called notebooks. Jupyter Notebooks is one of these examples. It's a Word document with text, but also you can have data behind code to manipulate the data, charts, which is powerful because on top of the document you have everything you need to build things bigger. And I will finish with one example that we are doing in Foundation 29. This is a non-profit organization that I founded a couple of years ago, which is the X29, which is a tool to support the process of diagnosis. The process of diagnosis for rare diseases is quite straightforward from a theoretical point of view. Is you have to identify the symptoms, you have to identify the genes, and you have to make the connections. But the thing is, this process takes days for a human. What we are doing in the X29 is use machine learning to speed up that process. And right now we have a model to identify tests to do advanced phenotyping. We have another model to rank the different mutations from the genome of the patient. This is a random walk algorithm. And we have a new, let's say, beautiful way of symptoms that the physicians can use to speed up the process. And at the end, the physician has suggestion of potential diagnosis. The thing is, this model is for diagnosis, but can be very useful to identify patients for clinical trials as well. Because at the end, diagnosis is about clustering. The challenges, and I guess this is interesting, it's going to be very interesting for the people. Despite of all the technology, I use a super powerful computer, computer is doing the basic task of machine learning, of identifying dogs and cats. In order to have 98% of accuracy, you have to invest a lot of. But if they are able to do with 100% accuracy when they are 2 years old. Some example of cats. So we have to learn from how the humans are doing machine learning, because it's really different than the way we The other problem is training bias. The models that we are generating are very, very affected by that. The model will be wrong. And this is one example from one recruiting tool that Amazon did some years ago. And the tool was recruiting people. So as you can imagine, if you are training the model of men, the women will have less probability selected. Another problem is something called adversarial attacks, and this is when you have this type of is easy, fake the model. And this is one example of just using these special glasses. The people can be confused like Mila Jovovich, because the glasses on these course are able to train the model, to use the model in a different way and produce mistakes. And this is an example with glasses, but you find an example with medical image to change the way the model is working. At the end, when you are working with machine learning, it's about transparency and predictability. Explain what the model is going to produce and explain how the model is creating those results. And these principles are very important for most of the machine learning, and this is something that we have to preserve. We want to ensure that technology will be used in the future. Thank you very much for your time.
Thank you very much, Julian. Elizabeth, I think you turn the agenda back to you.
Actually, I'll just throw it immediately to Alan. So you might have seen in the chat that we skipped the break. So in order to make sure that we have time to hear the presentations and possibly also have a Q&A session after the next two sessions. So that's why. So people will have to deal with the coffee breaks and bio breaks as we go along. And Alan, back to you.
Yes, my name's Alan Yeomans. I'm the moderator for the 5th session. We will be looking at things in a little bit more detail, a bit more technical. So we have 3 speakers for you today. Each of them will be speaking for about 20 minutes, and we'll have a short 10-minute Q&A at the end of each presentation. And we'd like you to keep your questions for the presenter who's just presented. So, and I'll be timing the presentation. So Dimitris did a pretty good job of almost getting us back on track, but I'll make sure that we don't lose any more time than we already have. I'll introduce each speaker at the start. The first speaker for us today is going to be Melissa. And Melissa has over 25 years of experience in the life science industry. She has degrees in mathematics and a master's degree in computer science. And she's head of the CDASH team at CDISC. So a lot of experience, and I'll leave it over to you now. And the name of your presentation is Designing a Risk-Based Validation Strategy. So over to you, Melissa.
Thanks, Alan. Cornelia, you're controlling the slides, yes? Okay, good afternoon, everyone. Lots of really great examples and case studies within Dimitris' session. I'm going to actually build upon the case study that was presented at the beginning of Dimitris' session relative to the Smart Data Query project. You can go to the next slide. So Prasanna and Ashley presented the Smart Data Query. I'm sorry, I'm just going to turn my camera off. Prasanna and Ashley described the Smart Data Query project, and I'm going to talk about how we designed and documented a risk-based validation strategy around that. I think one of the most difficult, aside from the technology and building the artificial intelligence and machine learning models and making that work correctly within the different, within the context of the different projects that we talked about previously, is really about making sure in a regulated environment how we can clearly demonstrate that what we've built is appropriate and that it is validated to produce the results that we expect it to be producing. So all risk-based activity starts, obviously, with a risk assessment, and it's really critical, I think, with respect to validating a machine learning algorithm within the context of clinical research, to take the time to perform that evaluation of the risks and determine the organizational comfort level or the acceptability of those risks being realized. This really isn't any different than what we do every day within the context of other areas of our clinical research. However, it is, a little bit outside of the box, given that machine learning is new to a lot of us within the clinical trial space. So when we look at the Smart Data Query implementation and our risk analysis, we revealed 3 different primary risks that were associated with using SDQ to generate queries to find data anomalies. And those risks were that a discrepancy would go unnoticed, so we would not query something that really needed to be corrected within the data. The algorithm could create an invalid query, or the algorithm could create nonsensical query text. And when we took a look at that evaluation of our risks, we revealed an acceptable mitigation or no meaningful additional risk from current practices. So when we talked through each of those different risks, we said, okay, well, clinical trial data, the first risk that we were concerned about was whether we would miss something in the data that might affect patient safety. And when you take a step back, the data that's coming in on the case report forms is typically transcribed from medical records, and therefore it's not really involved in point-of-care medical decision-making. Now, we want to maintain the ALCOA principles that were discussed in an earlier presentation, so we want the queries to be going out in a timely fashion, but if they don't go out in a timely fashion, they don't impact patient care, right? So again, if a discrepancy went unnoticed or an invalid query went out, we're not going to jeopardize patient safety. Our current practices also include humans in the loop. So some of data review is automated. Some of it, however, is done based on listings or outputs of data and applying medical knowledge to what has come in. And so because humans are in that loop already, sometimes queries already get missed. Sometimes, a reviewer sends a wrong query accidentally, et cetera. And so there were already very well-defined mitigations in place should a query be missed or should a invalid query go to the site. They know how to respond to that without taking data action. And any critically wrong data has multiple redundancies built into our cross-functional processes. So again, looking at those 3 risks that we identified with using AI in this application and machine learning in this application, we felt pretty secure with the level of risk that we were assuming. And, when we sit down and look at our validation strategy, which I'll go into detail on, we felt like the overall risk assessment picture for SDQ was fit for purpose. Next slide. So in order to understand the validation approach, I think we need to really understand what the high-level architecture for Smart Data Query looks like. So this is going into a little bit more detail than what Prasanna and Ashley discussed in their earlier presentation. So if you start out on the left-hand side of this slide, our data is coming in from our data repository. So we have at Pfizer, as I'm sure all the companies do, we have a lot of data stores, not just the individual study that we're working on, but also pre-existing clinical trial data that can really help us target and train the initial version of the machine learning algorithm and the model. So the data comes in on that left-hand side through an API into our machine learning pipeline, which is in the red dotted box in the middle of the slide. Once the queries are identified, the data is then fed through to an interface moving down into the bottom portion of the slide, into an interface where trained data scientists can evaluate whether a query being generated by SDQ should be issued or passed through to the data management system and eventually the site. So the decisions, Ashley covered a little bit of the types of decisions that are made. We can identify where there's a good prediction, and the query should go back into the pipeline and pass all the way through to the right-hand side of the slide in the upper portion to the data management system to be issued to the site. We could have a good prediction, but perhaps somebody has already raised that query. Perhaps that prediction or that query belongs in another category. So it's good, and we want the machine learning model to understand that it's good, but we're still not going to pass it all the way through to the data management system. And then, obviously, we could have a completely nonsensical query, and that information needs to be fed back to the machine learning model so that it does not come up again. So throughout all of that, of those decisions, those interactions, in the bottom right-hand side of the red box, the SDQ database is keeping track of the model performance accuracy. So it's taking the information that it's getting from the data manager interface, and it's taking those decisions on board, and it's constantly able to reconfigure the accuracy of the model performance. Next slide. So understanding the architecture here, what does normal look like, right? So sitting down to do normal, quote-unquote normal or traditional software validation, what things look and feel like what I'm used to, and what things need maybe a little bit of, different type of thinking. So first, the things that look the same and feel the same and are more or less validated the same is the data flow, right? We're able to, as data comes in to our machine learning pipeline and as decisions flow out of our data pipeline into the data management system, we can document and demonstrate very clearly the reliability of that information to come in correctly and to leave and arrive correctly. So data in is equal to data out. That is very straightforward validation paradigm, which many of us are used to with system interoperability. Our machine, we can also, even though it is somewhat complicated, computer science and math, we can verify and demonstrate that our machine learning model is generating the structure of the trees or the confusion matrix, the network correctly, right? So we know that the programmer, the software engineer that's defined the algorithm and the model that they've done that correctly, that the decisions, for example, in SDQ that are coming from our humans in the loop are being processed correctly by the code, and that we have training and test data sets that are maintained and traceable. We also have human decisions that are maintained and traceable. So again, that's a little bit of what data is coming back, meaning the decisions that the data managers are making, and then the utilization of those decisions in the context of the overall algorithm. And then lastly here, all of the traditional security, access, account management, audit trails, all of that validation looks very much like it would today in a traditional non-machine learning implementation. So we verify that the accounts are set up, that individuals provided with accounts are only able to do the actions that they're supposed to be able to do, and nothing that they shouldn't be able to do. And the system itself is maintaining a traceable audit trail of the different decisions and actions that are being taken within the system. So all of that looks really great, and that it's stuff that we're comfortable with. So far, we're so good, but if we go to the next slide, what looks different with SDQ? and where does that level of comfort begin to sort of disintegrate for the individuals that are typically involved in doing system validation and documenting system validation? Well, first, it becomes really difficult to write a script based on a data set, right? So we have training data sets and we have test data sets that tell us that the algorithm is producing reliable results. Normally, when we perform validation in a query system type of environment, we are able to prospectively identify and document good data or actions, and then bad data and actions, and be able to predict what that expected result looks like. That becomes completely non-manageable with a data set of information, so processing, gigabytes of data through a machine learning algorithm. So scripts look and feel different, and being able to kind of imagine that data set becomes very difficult. Also, with machine learning, we know that things are going to be wrong. And while we mitigate the level of items that are wrong using the accuracy rate and other items like that in the test data set, we still have humans in that decision-making process, right? So the data managers, we're still relying on the data managers or the data scientists to make valid decisions about whether a query is right or wrong and whether that query should be issued. And the model, as we know, will build itself the way that it needs to. It'll build the nodes in the network and manage the nodes in the network based on the feedback that it's getting from the humans in the loop. But inaccuracies are going to work their way into the training data set because a data manager makes a wrong decision on a wrong day, and then the model itself starts to skew. So we need to be comfortable with that. And that's a very foreign feeling for individuals in the clinical trial regulated world to get comfortable with. And then lastly, documenting what's going on in the trees, and I use that kind of in context, right, because SDQ is a deep learning, a semi-supervised deep learning implementation. So it's not really a tree, but it helps to kind of visualize, I think, for folks in having the conversation of a tree structure. We don't necessarily know or manage or want to know or manage the intermediate learning iterations of the machine model. That becomes not value add when we have the overall context of the training data set and the test data set that we can use to reliably recalculate our accuracy rate. So these are all things that become very uncomfortable when you're sitting down in a traditional, system validation with a system validation mindset in a regulated environment. Next slide. So in summary, our risk-based validation strategy for SDQ was really informed by that earlier risk analysis and risk evaluation that we performed at the beginning, right? So we wanted to know what are our errors, what errors do we need to mitigate against, and where are we comfortable with an error that might introduce itself. We started off with a very basic and standard software development lifecycle, right? So we verified that all of the key principles of that traditional SDLC were in place and were followed. So we have clear specifications for SDQ, what's supposed to be occurring, the integrations that the system itself has, the overall system it has, making sure that those integrations are tested, that the security and the access is tested for the different user accounts, and that all of the expected evidence around those pieces is stored. Next, we looked at, how we're monitoring that performance accuracy and how do we really document the data that's coming in, the training data, the the test data, and how we can communicate all of those things, really contributing to the overall reliability of the machine learning model. Well, this is where we kind of start to diverge, I think, from what we would normally expect in a true system validation in the world that we live in today. And really, when we took a step back, again, using our risk analysis and risk validation, we found that it was important and critical to have a written process with evidence of adherence to that process, right? So when we need to, when we hit a accuracy rate that is producing more invalid queries than correct queries, we need to make sure that there's evidence of adherence to monitoring that accuracy rate and then kicking off a relearn cycle. We also found that it's really important to keep the training data sets and tie those training data sets to the software version, right? So part of the validation package or part of the software package itself includes the training data set that was used to develop that model and release that iteration of Smart Data Query. And all of that gets Yep.
Okay. Good.
What's that, Alan?
2-minute warning.
Oh, okay, thank you. And all of that gets archived as software system validation, and we can reproduce that if needed in an audit or an inspection. And then lastly, but not leastly, it's always critical to invest in our staff, the humans, making sure that we have evidence of training and that training advises them of their part in the software decisions. I think this is really critically important because over time, highly skilled workers get used to software giving them reliable results every single time, right? So in a machine learning, artificial intelligence type of project, they need to know that the machine every now and again will return something to them that's invalid. And so it's important for them to understand the impact of their decisions in the overall accuracy of that model. It's a slightly different take, but absolutely critical in training staff that are empowered to interact with the decisions coming out of SDQ. So in conclusion, we're several cycles in with Smart Data Query, so several learning cycles. We're expanding the number of studies that we're using this application in. We're definitely starting to realize efficiency in what's traditionally a very manual and labor-intensive check, and our staff and our studies are benefiting from SDQ. Thank you. Take questions, Alan, I think?
Yeah, I can't see that anybody's raised their hands yet, but I had a you mentioned early in your presentation, or all the way through your presentation, that you're saving the training sets that you've used to train your SDQ and to validate it. I also thought I heard in one of the slides that you, when you get a negative prediction, you're feeding that back to the system, so that it won't make the same sort of negative query again. I was wondering, how does that work together with the training sets?
So the decision, what will happen is when the negative response comes back, the software will learn that within the network that the data flow through the decision-making nodes in the network is no longer a valid pathway, and so that pathway gets destroyed. With respect to the training data set, the training data set stays where it is. Actually, Prasanna, if you're still on, this is like starting to get a little bit deeper into coding than I normally get with validation. But those decisions then destroy the pathway that gets to that decision, and so the training data set gets updated so that pathway is no longer a valid pathway, and is maintained as we move across the models. So I know that's like sort of high level, sort of lower level, but
yeah, and this is
very
done
complicated.
During the time that the system's in use then. So it's like a continual learning type of aspect.
Yeah, the training data set builds up until you get to a relearning cycle, right? And then the model goes through a learning cycle, and you get a new training data set out of that.
Okay, that sounds very good. Does anyone else have any questions? No? Well, then maybe we can move on to our next presentation. Thank you very much, Melissa, for a very interesting
Thank you, Alan.
Yeah. Our next presenter is Bruno Boulanger, who's Chief Scientific Officer at PharmaLex in Belgium, and also a lecturer at the School of Pharmacy at the University de Liège in Belgium, and a member of the Committee of Expert Statistics of the U.S. Pharmacopoeia. He's got degrees in statistics from both the University of Minnesota in the U.S. and the Université Catholique de Louvain in Belgium, and he's been working for 25 years in several areas of pharmaceutical research. And he is here today to talk to us about opportunities and risks in using AI for discovery and development of new treatments, a Bayesian perspective. Bruno, are you with us?
So thank you for the invitation about this very important topic, and I probably try to give also another perspective regarding this general problem. So next slide. So the agenda, I've got 5 topics there, is probably to discuss about an important topic that also applies to AI solution, which is the problem of replicability, reproducibility. There is also a discussion about the amount and the number of data required, and I'm going to show that the more data we have, the higher the risk to have wrong decision. We'll try to see how can AI plug into a decision-making process and what are the risks, and insisting on the fact that we need to be very careful about the way we train AI solution. But this being said, there is quite a lot of huge opportunity, as we can see. So replicability and reproducibility are really growing issue right now in the literature, and AI will fall into this problem as well. So just to remind a recent definition by national academies, reproducibility is defined as obtaining consistent results using the same data and as original study. And replicability is having same result if we do different studies. And AI is falling in both categories. In the first category, I have to say, which is a reproducibility issue. Next slide. Just to remind here is that even with well-established control and known methods that are applied to the data, like classical statistic and frequency statistic using hypothesis testing, which has been successful in the past, there is no recognition that even well-established method like p-value, for example, is producing and raising concern regarding the reproducibility and replicability. And even if those methods, this type of problem is occurring, then we have an additional reason to pay specific attention to the upcoming AI solution. Next slide. Something which is very well known and that can be observed is that when you are investigating a large number of data and opportunities and so on, what we can see through the history is that the number of wrong decisions that is made is growing with the number of data we are collecting, the number of experiments we perform, and so on. And that's one of the reasons we start to see something which is brand new right now. Since 10 years now, it is the reproducibility issue. Not because there is something wrong with the way we make the analysis of the data, but also because we massively increase the number of data and analysis on the data we make. And therefore, with the big data and AI coming on board, we need to be clear that this issue of probability and the absolute amount of wrong decision may also grow proportionally to the amount of data. And it is something that is going to happen right now. So let's be conscious about this, even if there are really important opportunities behind this. So how to control that, and I think that the purpose of this meeting is to ensure that we will have a good control risk assessment practice to not fall into past traps that was already observed. Next slide. So as I mentioned, reproducibility is an issue that applies also to artificial intelligence. And there has been, back in 2018 or 17, 18, yes, there has been already some conference on the topic indicating that problem of reproducibility apply to the AI and machine learning. And there is no reason that there is going to be a change with that respect, because it is a measurement system that needs to provide some support of decision making. Thank you. Next slide. There is also a lot of debate about the quality of the data that are going to be used and to support this type. Quality data is really necessary. There is no discussion about that. But we need to be conscious that these are not sufficient. We need quality data, certainly, because if we feed the system with not appropriate data, we will not maybe have appropriate result. But let's keep in mind that historically, the control clinical trial that has been very successful and that allow major discovery are performed in very well controlled conditions with high quality data. But there is nevertheless always an issue, there are always a wrong decision that are made based on this type of very good quality data. So quality data is necessary, but let's be conscious that we need more than that. We need to have a more refined analysis, interpretation, and inference that we draw from those quality data. So that's an opportunity for AI here, but also a risk if we don't do that prudently. And I will try to show you some results. Next slide. So next slide again. So the idea is a problem of decision making, and let's assume that, and we have seen in previous slide, accuracy of a diagnostics of a solution is as follows. You know the concept of sensitivity and specificity, and let's take the example. What the sensitivity is the probability that your device, your diagnostic device, is going to provide you a positive result if you have a cancer, and the specificity is the other way around. But in practice, what we're looking is the opposite to this plan. So next slide. Meaning that we are interested what is the probability I do have a cancer given I receive a positive result. Next slide. So next slide. Take an example which is drawn from the literature, and here assume that you have a diagnostic test which is pretty good, with a good sensitivity of 86% and good specificity of 88%. If you test a large number of women, say hundreds of women here for sake of simplicity, you may end up with this type of result. Next slide. And what we can see is that if you got a positive result, there is, as you can see, one chance out of 12 plus 1, so meaning one chance out of 13 that you truly have the cancer if the lab is sending you back a positive result. So you see the probability is very low in this case. Next slide. And next slide. And this is the fact that the probability is very low while we have a very good sensitivity and specificity is linked to the fact that the prevalence or the event of interest is low in this type of study. Say, for example, 1%. So that's very important to keep in mind that there are 3 P's that are supporting the decision-making process when we use a solution. Sensitivity, specificity, of course, but also, as we will see, the unknown or more or less known prevalence or probability of occurrence. Next slide. So, will artificial intelligence allow us to de-risk decision-making? So, assume that we have an excellent solution that is coming on board, and in our previous example, we are able to boost the sensitivity and the specificity of our diagnostics test. And we move from 86, 88 to 95% using AI. In that case, as you can see, the probability that I have a cancer if I receive a positive result from the lab is moving from 7% to 16%. So the probability is still remaining very low. Why? Because as I indicated previously, in this scenario, the key decision, the key aspect, the key piece is the rarity or the prevalence of the problem to detect. But let's keep in mind that we are going to use artificial intelligence in a lot of scenarios, but not to be able to detect what I call the elephant in the room. We are going to use that to detect and try to identify rare events or rare patterns that are going to be embedded into the noise. And so the rarity or the low prevalence, the prior prevalence is still something very important that we need to keep in mind. We are going to evaluate the ability of AI solution to provide and to support efficient decision making. Next slide. There is another aspect also that apply to different practice, and particularly artificial intelligence in this case. For example, you know that if you are going to conduct several clinical trials on the same drug, the same treatment, for example, on this type of meta-analysis, you can see in the literature that the result you will get from one trial to another is going to vary slightly, and sometimes you may have some contradiction, but globally you will see some variability, and that's something very important. Keep in mind that all those trials are performed in very tightly controlled and homogeneous conditions. We need to have something which is precise and biased. Okay? But what you can see that even in those very controlled conditions, we got always different results from one study to the other. And so that's something very important if we want to train artificial and machine learning solution to ensure that it will be as large as possible. And so, next slide. Yeah. This is assume you may have different type scenario where there is either on the top left no variability from trial to trial, and you can see the control and the treatment group are always whatever you may have, or you may have a scenario like in the bottom line where there is a study-to-study variability. The treatment will show different results, and there is therefore a difference between the two treatments from one study to the other. This is what you may observe if you have the opportunity to conduct a large number of studies, which is usually not the case. Therefore, what you are going to do is maybe to conduct only 1 or 2 studies. Next slide. And so assume that we take at random 2 studies out of the many studies you could potentially do. As you can see, because of the study-to-study variability, which is quite important, you may or you may not have a difference between the 2 treatments. Okay? This is linked to the impact. And so there is a confounding between the study effect and the treatment effect, which is something very important. This issue particularly applies to classical approach, and therefore, if it applies to classical approach, is going to have an impact on artificial intelligence because it is not linked to the, I would say, the insight intelligence of the solution. It is linked to the design of the study that will provide data to feed this intelligence. So the problem remains. Next slide. And as we can show, and it is something you can find in the literature, if you have quite an important study-to-study variability, which is what we observe in general, what we can show that by ignoring this piece in the training part or in the design to train this type of solution, you take the risk to considerably inflate your type 1 error. You got the feeling because you work in very well-controlled condition that you have very nice data, but unconsciously, if I dare to say, in a way, you increase the risk of false discovery, which raises to the issue we know, which is the major crisis in terms of reproducibility and replicability. Next slide. Next slide. And so the idea is you start to work with artificial deep learning network, for example. What may happen is that, assuming that you have what I call a clinical landscape in general, if you train in one specific set of conditions, in one specific area, and so on, what may happen is that your AI solution, maybe from a technical point of view, will be very reproducible, but it will be trapped into a specific dynamic, just represented here by a strange attractor. And therefore, the outcome of this type of solution will depend on the place where it has been trained. And that's an issue that has already been mentioned by several observers when we use, for example, artificial intelligence on the social network. I mean, they are trapped by what they've been exposed to. And so it's very important for the future to ensure that we are going to maximize the diversity and the source of variability to train the AI solution. This is what we call a quality by design type of practice, is to ensure the solution and the decision making it will provide is going to be robust, whatever the location and where we locate into the clinical landscape at large. Next slide. This being said, there are massive opportunities, and here I take a theoretical example we've been working on, is, for example, in the development of new treatment for Parkinson's disease. And we know that sometimes it may be challenging to follow the real evolution of those patients because we see them from occasion to occasion, and some of the scale, like UPDRS, are, for example, extremely variable because it depends on the day, the condition, and so on. And so using wearable device, as long as they are fully qualified, can give extremely valuable information about the progression of the patient, and therefore it may have some important impact on the ability to conduct a study for new treatment in Parkinson's disease with smaller sample size, and therefore maximizing the chance and opportunity to discover a new treatment for such a disease, for example. Next slide. So the conclusion is More data doesn't mean less risk. Something that we know and we learn right now is that the more even quality data, the more wrong decision we make. Let's be careful of not repeating some of the error that has been done logically in the past with omics and repeating that with the current digital world and artificial intelligence. We may have the feeling that we are going to improve drastically the decision making by increasing the performance and accuracy of AI, but improving sensitivity and specificity, as we may see, is not always worse, and it will depend largely on prior scientific knowledge and on assumption in some cases. It's very important to take care of the way we are going to design the study to train artificial intelligence and to test them as well. But this being said, if we pay attention to those details, the opportunities for unmet needs are really outstanding as long as we are prudent, but there is clearly an open route. So thank you for your attention.
Thank you very much, Bruno. And you were right on time as well. You went into your conclusion slide just as I was going to give you your 2-minute warning, so you couldn't have been better on time. Does anyone have any questions for Bruno? I can't see any raised hands in the program, but I don't know if that's just me. Do we have any questions for Bruno?
There's a question from, let's see, oh, that disappeared again, sorry.
Okay. I must admit, statistics is not my area of expertise, so I'm not going to make a fool of myself by trying to think of a question for you, Bruno. So is there something you would like to say that you often get asked that you think is relevant? Nee has a question. Someone is writing there.
Yeah. Hi, Bruno. Thanks for your presentation. Based on my therapeutic area expertise, I see there are a lot of therapeutic indications that there are a lot of trial-to-trial variability. So if I understand your presentation correctly, before pooling any data, for example, like if somebody wants to pool all the placebo data to have as a control arm, if there are if the trial-to-trial variability is very high for those therapeutic areas, we should not pool data and come up with a pool control arm. Am I correct?
I think it's very important to figure out what is the trial-to-trial variability and to take that into account. Okay? Because if you don't pull all those data, you may end up with a bias. Okay? So it's very important to combine all this information, for example, about the control group, and to integrate that into the VIR control group or the synthetic control group, to ensure that you are going to carry on to study variability. Otherwise, you may end up with a biased conclusion.
But if they want to use the pool average data, let's say as the placebo data, instead of having a placebo control arm, and in therapeutic areas, for example, like antidepressant trial, statistical differences between drug and placebo is just 2 points in commonly used rating scales. So, and some of the trials, placebo arm variability can be quite wide, more than 2 points. So something like that, I would, if I understand you correctly, we should plot and see how variable trial-to-trial data is like first as somebody attempts to pull the trial data.
I think if you have the data from several trials, you better have to combine them to have an idea about what is potentially the trial-to-trial variability. That's very important. Yes.
Okay.
Because it's going to be
so
variable. So you protect yourself against a lack of reproducibility doing so.
Okay, so basically explore both ways, look at the trial-to-trial variability, and then combine and look at the how
Yes.
the data will look like. Okay, thanks.
Yeah, important, yes. Otherwise, you will make your type 1 error by ignoring this. Yeah. The same applies to AI, by the way.
Right. Thank you.
Thank you.
Okay, there was a little bit of chat going on in the background here in the chat session, but I can't see that there was actually a question in it. Just seem to be people agreeing with you, Bruno. So if there are no more questions, then I think we will move on to Kevin. Thank you very much, Bruno, for a very good presentation.
Thank you. Thank you to all participants.
I'll just try to turn my camera back on again. Hello, still here? Yes. Okay. I'd like to introduce Kevin Lyman. He's the CEO at Enlitic, who work with artificial intelligence and deep learning. He's also got a degree in computer science. He's worked at SpaceX on the Falcon 9 rocket and the Dragon space capsule, among other things. He's also worked at Microsoft. And he's also the founder of the Inventors Guild, a team of students from 10 of the country's top universities who earn course credit and money by consulting for startups. And so today, Kevin is going to talk about the application of deep learning to medical image analysis for us, where deep learning technology goes further than simple heuristics, pattern recognition, or machine learning. Are you with us, Kevin?
Yes, I am. Are you able to hear me?
Yes, we are. Wonderful. Go for it.
Fantastic. Well, the first slide didn't have any text on it, so hopefully that rendering issue doesn't appear anywhere else, but we can move right on to the next slide. Today I've been, so first off, thank you all for having me, and thank you, Ellen, for the introduction. Today I've been asked to talk a little bit about continuous machine learning and as well as applications within medical imaging. And just prior to jumping into that, to give a little bit of background on Enlitic and who we are, we're a startup based in San Francisco that has been around for about 6 years, and we use artificial intelligence to develop clinical applications for primarily radiology, where the goal is not to replace clinicians, but rather build tools that sit alongside them and enable them to provide faster, earlier, and more accurate diagnostics. We do look at a few other areas of digital diagnostics as well, cardiology, pathology, et cetera, but most of our focus is radiology. And we do a bit of work with pharma companies and insurance providers as well, doing things like stratifying risk or identifying patients for orphan trials. But our core focus is really augmentative software for workflow for radiology. In the time we've been around, we've been fortunate to be recognized a number of times for our work in the space, most notably being named to MIT Technology Review's 50 Smartest Companies 2 years in a row, and more recently, next slide, having won the first CUBE Global Tech Fair, where Steve Wozniak, the technical co-founder of Apple, gave us the €1 million grand prize, as well as this wonderful quote, which I feel is quite relevant for our discussion today. In my opinion, Enlitic is what technology should be about. We should trust our machines to be smart. And this is sort of the ethos that we try to live by, recognizing that human-centric design and validation are arguably even more important than the engineering and technology that goes into building the models in the first place, in that there's really very little reason to have a model that gets 99% accuracy if nobody trusts it enough to actually work alongside it and enable it to influence their outcomes in a positive direction. Next slide. To start off with a more concrete and understandable or intuitive example of the type of work that we do, the first model that we ever built and deployed was wrist fracture detection, a picture of which you can see here. And this was about 4 or 5 years ago. The way that this application works, a radiologist can bring up an image of a wrist x-ray in whatever software they already happen to be using clinically, And automatically, these blue circles appear on the image, highlighting any regions of interest that the underlying algorithm believes may correspond to a fracture. Now, in order to prove that this system works, I used to be on the modeling team at Enlytic before becoming CEO, and it was actually my job to run the study on this to validate that it was performing as expected. And of course, we had done a great deal of internal validation work, but to really prove that it works in a more live environment, we ran a trial with Capital Radiology in Australia, where we had 6 of their radiologists interpreting each of 400 wrist x-rays, once with help from the algorithm, as in seeing these blue circles, the other time without help, so blinded to the fact that an AI had even looked at it, and with 2 months of washout period in between to ensure that they forgot what they saw, and randomized as to whether the first time had help or it was the second time. We then followed up each of those patients with a CT or an MRI to act as a gold standard ground truth follow-up for did these patients actually have a fracture or not. So consulting a modality with much higher specificity than the plain film in the first place. And what we managed to show through this exercise was a few things. First, that radiologists with our help were reading cases 21% faster. Second, they were catching 11% more of the true positive fractures. And lastly, they were making 9% fewer false positives along the way, all of which were very promising findings and had indicated to us that, yes, AI can be helpful to radiologists, at least at the task of detection of fractures. But what it also highlighted was, unnervingly, a couple of issues that stand in the way of practical success for a commercial organization with limited resource. First, that ultimately radiologists are on the hook to look at any part of the human body from any angle in 2, 3, or 4 dimensions. And regardless of the scenario, they have to identify every abnormality, even if it's something no one's ever seen before. So how much utility are they really going to get from something that just points out wrist fractures? We may save them 21% of their time, but on a study type that they spend less than 1% of their time on. The other thing that it highlighted was that it took us about 12 months of dedicated effort on development and validation for this algorithm that could identify one finding in one type of scan. And so this really highlighted that to really be successful here, we need to invest in exponentially growing the number of outputs or indications or claims that this technology can actually help clinicians with. But second, we need to exponentially scale the rate at which we can develop these. And that's really where we started to take a look into more continuous learning methodologies. Next slide. Now, to talk about a very different type of application that really explains why this focus on continuous learning becomes much more necessary, rather than looking at wrist x-rays, now let's look at chest x-rays. Chest x-rays are the most common type of radiology study done anywhere in the world. They make up probably about 40% of radiology images taken worldwide. And this is because they're an incredible screening tool. They're highly sensitive with signs showing of a number of different conditions. The flip side, however, is that they're not very specific. It's very challenging to tell what exactly is wrong, even if it's relatively straightforward to tell that something is wrong. Now, not wanting to fall into the same trap on this that we did with wrist fractures, right off the bat, rather than trying to teach a model to find one thing in that image, we sort of took the opposite approach. Let's train the model to figure out that the image is abnormal for some reason. And the way that we did that was by showing the model as a baseline, essentially 1 million chest x-rays that were comprised from Australia, the United States, all over Europe, South America, Asia, and Africa, so a very diverse set. And all we told the model was which half were normal and which half were abnormal without saying why. And we found that that resulting model was actually quite good as a result of triaging normal from abnormal. But the problem it had was that it didn't really know why anything was abnormal. And this image that you see here on the right is the cover of Applied Radiology from July 2018. And the cover image was actually generated as one of our model outputs. That's the result of our algorithm at the time, reading a chest x-ray from one of our partner sites and highlighting that this image is abnormal because these two regions have something wrong with them. It happens to be highlighting, in this case, a nodule projected over the right mid-zone, as well as a linear atelectasis in the lower left lung. And that said, the model didn't know that that's what those two things were. It simply knew that those things shouldn't be there. But if we look at where the model is at today, that same model not only is considerably better at identifying what's abnormal, but now it can actually articulate why it's abnormal with more than 50 different kinds of findings that it can actually indicate what the issue is. And probably the most interesting aspect was that this required considerably less hands-on effort than it did to build just fractures in just wrist x-rays. And that's through this investment in more continuous methodologies. Next slide. If we take a look under the hood of that chest x-ray model, there are so many things I can identify that I actually couldn't fit it on a slide. I tried to, but I already had to make it as small and unreadable as the section you see over there on the right-hand side. And that's really to give you a sense of how much more sophisticated this is now than a model that just determines normal from abnormal. Beyond that, it's additionally able to articulate the difference between normal studies and unremarkable studies. The difference there being that there are anatomic variants or findings that are very common that are not normal. They shouldn't necessarily be there, but they're also not remarkable, as in they're not clinically significant or actionable. Now, to touch into why we needed to look toward this continuing learning methodology, I think when most people think of continuous learning, it becomes very concerning very quickly in that it gets mischaracterized as autopilot. Really, continuous learning is not autopilot. It's far more akin to cruise control. I need to keep my hands on the wheel. I need to keep my eyes on the road, but sometimes I can take my foot off the pedal unless the speed needs to change. And that's really how I would equate continuous learning, in that we're really automating a lot of the systems that go into model training, development, validation, and deployment, which I'll discuss the mechanics of momentarily. But that enables us to take a step back and look at the big picture of, are we validating this appropriately? Are we collecting data from enough different diverse populations? Are we validating the different types of tasks this is performing in the appropriately different ways, whether it's just doing image level categorization versus drawing an actual ROI on the image versus providing a characterization or a measurement. Each of those are different, but it becomes very challenging to focus on both the day-to-day tactics of moving data around, as well as the big picture strategy of, is this even learning the right thing in the right way? And continuous learning is really a way to move toward the latter, focusing on the big picture while understanding that a lot of the underlying mechanics are the same thing, just happening in a more mechanical fashion. Now, while the way that many people think of the primary use of continuous learning being to just improve model accuracy, in reality, it enables you to take a practical approach to a lot of problems that would otherwise be unapproachable in the context of using machine learning. The two obvious examples that people think of, again, being just naively improving the accuracy on a category that's already recognized, and second, reducing errors in known categories of error types. But where we would look at one of the most interesting aspects of continuous learning is this ability to build depth and breadth to an ontology. Now, as I had indicated earlier, when we first built this model, all it could recognize was normal from abnormal. But at that point, we look at it like a binary decision tree, where the goal is to get more specific on what abnormal means, while we focus on looking at model outputs in order to help it along the way to categorize what that means. Each of the bold and italicized rows that you see in the table here, such as pulmonary parenchymal abnormality, pleural abnormality, mediastinum abnormality, you can think of that as the next level of the tree, that the next round of iteration, the model pointed out those high-level categories. But then by the third time around, it started to break that down, each of those categories, into more specific children. So pleural becomes pleural fluid, pneumothorax, other pleural, pleural mass, etc. Now, this also has the benefit of enabling us to much more effectively generalize existing models to other problems in other body regions. We've actually, since taking this approach on chest X-ray, taken the exact same model, exact same infrastructure without changing anything, and simply pointed it to a labeled data set of fractures across various body regions in plain film. And it took about 2 weeks to reach near expert level performance, where it can now identify fractures of the spine, of the wrists, of the skull, etc., and with validated very high accuracy. I'll touch on the questions at the end, but there is a very good reason why we don't use the existing ontologies. Next slide. To touch on some of the infrastructure that enables this to happen, I just included here a screenshot of one of our architecture diagrams. And while we don't have all the systems in place that would really be necessary for an ideal continuous learning system, we do have most of the critical ones. And just to touch on a few of those, you can think of our infrastructure as having three primary layers to it: a front end, a back end, and a service layer in between. The back end, which you see along the bottom row, is really a glorified data lake where all of our clinical data gets put into, whether it's an image, a vocal waveform, text, regardless, all goes into Vault. The front end is what we use for visualizing results as well as capturing labeled information to go into the system. And the service layer is where most of the interesting things happen. That's where we semi-automate a lot of the development activities that would otherwise take a considerable amount of tedious resource. A good example would be our System Modeling as a Service, or MOS. That's a tool that automatically takes our models whenever they meet certain triggers and deploys them as APIs so that we don't need to worry about that deployment system. That additionally enables us to easily create complex pipelines of models by simply plugging them in with basic function calls. Another example would be the system you see to the left of that, Executioner, which I know is a great name for software in a medical device company, but that's our tool that semi-automates the training of models and indicates this is the hardware to use to train that model for this point in time. But I've laid out along the bottom here 4 high-level principles that I think are really critical to keep in mind moving toward a continuous machine learning process, especially in clinical applications. The first, which is most important, but hopefully is obvious, is that all of the data should be in one location. And I've broken that into 2 ways of looking at it. The first is that everything sourcing data needs to be putting it in one location. The second is that everyone requesting data should be looking in the same location. Now, this is important because, of course, one of the hallmarks of how the model is going to improve over time is by looking at new examples from new parts of the world with new pathologies involved and with new opinions from new clinicians looking at it. But if it has to look in a new location each time that it's doing that, the problem grows exponentially and becomes incredibly unmanageable. But if instead we have all of these processes consulting the same location, it enables us to more or less set it and forget it. As long as there's good rules about how that data is curated and indexed, we don't have to worry about moving data all over the place. We've already all agreed, next time you ask the piggy bank, it might have more money in it, but you're not asking a different piggy bank. You're asking the same one. The second high-level principle is that model retraining should be triggered by data monitoring. It shouldn't necessarily, and by that I mean there should be known triggers that indicate when we want to retrain a model, as in when this data set reaches twice the size it was before, or when we have 50% more labels of this particular type of finding, or when our accuracy hits this particular benchmark. All of those tend to be things that reasonably easily can have rules put around them, where it removes that necessary step of saying, I want to retrain this every time. And because we never throw away an old model, all of that's always available through version control, there's really very low cost to being reasonably liberal about when you want to kick off that retraining. As long as, the second point made on this item, those quantitative results should be calculated automatically and stored in a location where it can be easily visualized and acted upon. And I'll show an example momentarily of what many of these systems look like practically. But this is really critical to ensure that we're really in the driver's seat of not necessarily moving each mechanical action, but again, making sure that overall the process is heading in the right direction. The third point here is that our results, as in how well we're performing in each different category, should be used to prioritize whatever data we're then showing to humans to have them label or review the outputs of to ensure that the model's doing well. If we think about the giant list of outputs I showed earlier, well, through that continuous validation exercise, we know that it might have 0.72 AUC on lung nodules and 0.98 AUC on enlargement of the cardiac silhouette. Well, if that's the case, we really should be prioritizing cases to put the lung nodule examples up top and the cardiomegaly examples at the bottom, if we already know our performance is good enough in that category. Now, at the same time, we need to be making sure that our input into the labeling system is appropriate. That means assessing any labelers that go onto the system before they're allowed to touch any data to inform the model, and also regularly to ensure that their performance never dips. And lastly, as I started to indicate a bit earlier, we should be deploying these models automatically, but into sandbox environments where they don't necessarily pose any patient risk, but still put it into a natural environment for us to gather that simulated feedback. And our monitoring of this entire process should enable us to easily, with some basic interfaces, balance those hyperparameters that drive this process. Next slide. To talk a little bit more about the labeling aspect of this, this is an example with a couple of screenshots of our annotation platform. And there are two views that you can see here. The top view shows our dashboard, which is what radiologists see when they apply to work with us. And right away, they can see that there are lots of different types of data that we're currently labeling. And inherently, we needed to move in that direction because if we're building models not just for chest, across the full body, well, even if we're not building a model for it right now, there's no reason not to be labeling data and collecting that in the background so it's ready when we do need it. But you'll see that, and I apologize for the blurriness of the images here, but brain CT, head X-ray, abdomen X-ray, everything along the bottom, you can see that there's a button that's grayed out that says Take Test. And this is to represent that when you apply, we don't really care how good your credentials are. You're not allowed to label brain CTs until you've passed the test and proven to us that you know the difference between clinically reading and reading for the sake of training a machine learning model. After they pass that, we then enable them to label by clicking those buttons that appear that you'll see under lung oncology, chest X-ray. Along the bottom, those are buttons that say add 1 task, 5 tasks, 10 tasks, etc., so that people can simply choose what they'd like to label that day and then go through the list. Now, the bottom screen that you see there is an example of an individual labeling task. And in particular, this is what we call a general anomaly labeling task. We have, I'd say, probably a dozen fundamentally different types of labeling tasks for different types of data. But this is generally the one we start off with. And you can think of the task here as, if you see something, say something. We give a free-form text box with a drop-down. If they click the drop-down, they can see the full ontology we have of abnormalities we recognize, but they can also start to type in the box what they would call whatever finding they see. If they enter a new term we've never seen before, that then goes to our ontology management team to determine, is this something new or is this an alias for a phrase we already recognize? We find that this gives us maximal flexibility both in growing what the model can learn and letting the people who are training and validating it do the driving of what it should learn in the first place. Then we ask them to highlight on the image where the actual finding is. But to ensure that labelers remain good at this process, we quality assess them every week by ensuring that 10 studies are the same across all users in a given stream, and then having one of our managers internally, typically a radiologist with at least 10 years experience and subspecialty training in that particular area, will then go through and grade those cases weekly to indicate who gets to stay and who has to get banned from that list. Unbeknownst to the person doing the grading, 3 of the labelers that they're assessing are actually the best performing models of the week. And that feedback is used to drive who the winners are from a modeling perspective. So you can think about that as being sort of a mini Turing test that's built into our weekly validation exercises from a labeling perspective. Next slide.
Kevin, you're just about running out of time. Yeah.
Great. I'll just breeze through this then. So I had also mentioned that it's important to have a lot of this information from a validation perspective quantitatively calculated automatically and put into a dashboard for visualization. This is an example of our system we call Model Insights. For every model that gets built, it appears as a box on the top screen there where we give our users and ourselves internally an interactive visualization of all the models that are there and how they interact. Below that, by clicking any of those boxes, you can see the actual performance with full ROC curves. There's a little drop down there that you can use to train any of the different findings the model finds, since of course performance will be different on each. And if you zoom in real close, you'll actually see there's a bunch of blue dots on that graph. Those dots each represent a human who took the same test in order to show this is how well the model does at this particular task relative to a number of other radiologists. And interacting with those dots, you can see how many years experience they have, their subspecialty, et cetera. And the last thing that I'll just spend 30 seconds on, next slide, is starting to put these into environments where we can gather that feedback live and use it to drive the development of those models as well. There are really 2 ways we look at that. The first being retrospective, the other being prospective. we tend to take the mindset that if we're going to deploy something externally, we want to do it in the lowest risk environment possible and then progressively gate it into more high risk environments. Our lowest risk environment externally is retrospective insights, where we enable our users to see the outputs of our models, looking at all of their historic data, comparing their images to their reports to identify findings that may be discrepant in order to gain the business intelligence of what types of findings they miss, what radiologists are making mistakes. And inherently, this is a good example of a selfish interface for us in that we're pointing out discrepancies to radiologists. If we're correct, we're bringing them value in pointing out something they missed. If they're correct, they're providing us value in expressing where the model has gotten it wrong, but in a low-risk environment where there really isn't any damage that can come from using it in that way. And next slide. And once we know that it's performant enough in that kind of environment, only then would we put it into a prospective environment like you see here, where we actually have a full suite of live clinical software that for any algorithms that are approved and sufficiently validated, they go into that environment to help you read your live clinical cases. And even there's interfaces built in to disagree with us. Though in the more live clinical setting, we use a lot of information regarding should we trust that radiologist or not. We don't just blindly accept every correction as necessarily being correct without considering in the grand scheme of things. I'm sure I'm out of time now, and I know that was quite a bit of stuff, but I appreciate it, and happy to take any questions.
Yes, thank you. We've got one question in the chat. I don't know, Christian, if you'd like to put your microphone on and ask it yourself, rather than me asking it.
And I'm happy to touch on that one since I can see the question and acknowledged it. So there are a number of existing ontologies, SNOMED, ICD, etc. The problem is that they're very over the top, and they tend to be very inconsistent, but I would say inconsistent from the perspective of an external individual who is looking to repurpose it for something else. And it was, in SNOMED's case, put together for the particular purpose that it was meant to drive at the time. This means that there's a lot of categories and nodes on the tree that would conflict for our purposes. And the simplest way to put it would really be that it's much more, it includes a lot more nodes than realistically we would need to include for this purpose. And we found that it overwhelms a model if we start off that way. And rather building it up along the way, a more grounds up approach enables the model to converge much more quickly into intelligible subcategories of information. So it's a lot more work to build up the ontology, but we found that it's the only real practical approach to getting anywhere in a reasonable time frame.
Yes. Thank you, Kevin. This is a lot more complicated and a lot more detailed than just coming up with a standard training set and using that to train your AI. This is sort of very much continual training that just goes on and on.
I was just going to say that for that reason, it means a lot of the reason we've taken this approach is actually to de-emphasize a lot of the energy that has to go into what architecture do we choose, or how many forms of data augmentation are we applying, as well as a lot of the mundane day-to-day of where is that data set, and how do I consume this shape of label versus this other one. It's really eliminating, and by standardization, a lot of those issues so that you can focus your attention on the important part. Like, is my data set diverse enough? Is ground truth defined appropriately for the task that I'm actually trying to prove a claim around, et cetera?
Okay. Another question from Lisbeth here. Could you elaborate on what kind of documentation you would keep for documenting the validation or retraining that's being done?
Sure. Yeah, we part of the reason why we focused early on so much on making sure that all the data is in one place is because even if we didn't take this approach of continuous learning, it's good habit, and it forces you to, or heavily incentivizes you to apply a number of other methods that we would consider just good in-house quality. A good example there being data auditing and logging. Anytime any piece of data is touched by any actor on the system, meaning any model, any piece of software, any human being, a log is generated of that event along with the purpose for why that was touched. And so that's an example of some of the semi-automated documentation that goes into that. That all gets tied into our version control system. I typically like to do all this with live demos, but the model insights interface I showed earlier, if you scroll lower beneath that rock curve, you can actually see the full validation sets against which these were tested, and you can use that to access the full training sets as well. So not just internally, but externally, we allow people to kind of get under the hood and see all of that. And through version control, even see how that's kind of changed over time. Though for many users, it's a little overwhelming to look at that. But I would say that in general, it's the same documentation that would need to be put in place for any one-off application, like what I showed for wrist fracture detection at the start. This is just a method of recognizing where some of that documentation can be templatized and putting systems in place to record that information in the appropriate format. And I would think about it as if we're building a self-driving car, both the algorithms that enable the car to drive itself and the car itself need to be validated. But it's very painful if we need to validate the entire car and every component of the car every single time that we're validating a new algorithm. And so this is a way of doing very heavily validation at the start of each of those semi-automated systems, and then focusing more of your one-off attention on any of those individual algorithms that come in.
Great. And then finally, just to round off in 30 seconds, what would you like to see in the form of regulatory support for these types of systems?
I think the direction the U.S. is moving toward with the pre-cert program, I think, is incredibly forward-thinking, and I think it's necessary, given that I'm certain we're not the only ones using this kind of methodology. If you, without even necessarily thinking about the under the hood, how we're doing it, if you just look at that one slide with the number of outputs that we have in our chest X-ray model, that's going to be a big challenge for regulators for how are we going to validate each and every one of these 50 independent claims, and how are we going to continue monitoring that when there's improvement across any one of those categories. And I think the direction that the FDA has suggested with the pre-cert program is more to not to stop validating the claims themselves, but to put less emphasis on validation of every single claim, recognizing that 98% of their core is the same thing, and putting a lot more emphasis on validating the process and the infrastructure that enables that factory to run. So it's getting much, much closer to the company and their development process, and being more hands-off on each claim after claim after claim, as well as letters to file and things of that nature. So that direction, I think, is I don't really see how this will be sustainable without more countries moving in that direction. The other part is having more public validation sets. I think there is some danger to it, but seeing regulators move in the direction of Kaggle, I think, would be huge. For those that don't know, Kaggle is an online machine learning competition platform where companies can host data sets they want problems solved on, and the winner who built the best model gets money. There, they've managed to, even with money on the line, have a good system for hosting independent data sets and getting independent results that are known to not be biased from competitors around the world. If there were any kind of, even at an international level, system where regulators had recognized data sets that they withhold but do consistent testing on, I think that would enable everybody to go in this direction much, much more quickly.
Okay, thank you very much, Kevin. And I apologize to Ib, who had a question, but I'm being bugged that it's time to move on to the next session. So we've got our email addresses here. You can send any of us questions from the presentations, and we'd be happy to answer them. And I've been asked to just go straight into presenting the next session, which is going to be hosted or moderated by Torsten Stemle and Patrick Nadolny on perceived challenges and potential approaches by regulators and industry. So I'll hand over to them now. Thank you very much.
So thank you all. Welcome to the last session today, perceived challenges and potential approaches by regulator and industry. First of all, I'd like to thank Alan, Melissa, Bruno, and Kevin for a very insightful presentation on risk assessment and validation. It's a very nice way to set the theme for the next session. So we'll organize the session with 2 short presentations of 10 minutes each, followed up by a Q&A that Thorsten will be moderating. So our first presenter is Matthew Diamond. Matthew is a Chief Medical Officer at FDA CDER, and he even wrote a co-author of a proposed guidance by the FDA on the use of machine learning and AI for software as an equal device. And Matthew is going to share with us his insights about that regulation on best practices for developing ML and AI solutions. Matthew, the floor is yours.
Thank you very much. And just to clarify, I am at the Device Center at FDA, the Center for Devices and Radiological Health. I'm not at the Drug Center. And I'm located within the Division of Digital Health. Thanks for putting the slides here, and really thanks for the opportunity to speak. It's a pleasure to be part of this conference. You can advance the next slide, please. Okay, great. So I think this is great timing to be speaking on this topic and to be following Kevin Lyman's presentation, because the technology at Enlitic is a perfect example of the type of artificial intelligence, machine learning based medical device software that is our focus. And I think it has important ramifications for clinical trials for 2 primary reasons. One is these tools are being used in trials themselves, as previous speakers have mentioned, to analyze medical images, to analyze data from wearables, and I can share with you our approach that we're developing to oversee these products at CDRH. And the second reason is, more broadly, we feel that many of the approaches to these technologies, to their regulation, also have implications more broadly for all of the other AI applications for clinical trials that have also been discussed today, and including the idea of continuous learning that Kevin spoke about, and we'll talk about further, good machine learning practices, and other areas as well. And what I'm showing here are 2 other examples. At the top, you see the Caption Health software that we authorized earlier in the year, which assists healthcare practitioners that are not specialized in obtaining ultrasound images, but with this technology, they're able to obtain cardiac ultrasound images that can be used for making a diagnosis. So in the broad category of technology that assists a healthcare practitioner to do their job. And on the bottom is an example of a technology, IDXDR, that we authorized a few years ago. This technology is in a class of products that actually autonomously analyzes an image and takes the place of a healthcare practitioner in screening patients for certain types of diabetic retinopathy, and gives them access to this screening where they otherwise would not have that. So if you could go on to the next slide. I want to talk more about this continuous learning that we've already been discussing today. And we see this as a responsible form of innovation in AI/ML-based software that we would like to encourage and facilitate. And this diagram is actually taken from the International Medical Device Regulators Forum document on clinical evaluation of software as a medical device. And I'll walk you through it quickly. We've seen a number of similar cyclical diagrams today, but starting on the upper left with the definition statement that defines what a software as a medical device does, what is its intended use essentially, and as that device enters the market, if we follow clockwise around the circle, we gather new data. That data is analyzed. I'm speaking here from the perspective of the developer. And then with the generation of new clinical evidence, that device can essentially be used for a new purpose. And that definition statement for the intended use of that product can be revised. And I think Kevin spoke eloquently about how initially the technology that analyzed, I think it was the chest x-ray, initially could just identify that there was something wrong there, but with deployment in the field and the gathering of more data, it was actually able to provide a much more specific guidance and characterization of those lesions. And this is the type of responsible innovation that we would like to encourage. Next slide. So as we see an evolution of what was a traditional hardware-based device world to an evolving digital health device world from left to right in this diagram, we understand the need for a tailored approach that is scalable. We see product development timelines moving from months to years in a hardware-based environment to much shorter timescales, weeks to months. And we see much higher availability of post-market data that can be utilized, and the potential for really an exponential increase in the volume of these types of products, in large part due to the decreased barrier of entry to a software product than to something that was produced by traditional manufacturing. So what does a tailored approach, what would that look like? If you could advance to the next slide, please. And Kevin mentioned the PRECERT program, which is one of the pilots that are run out of our group at FDA. Essentially, we feel it's important to make these technologies available to patients, to do that as expeditiously as possible, including the modifications and improvements of them, of course, while maintaining a reasonable assurance of safety and effectiveness. We are looking for an approach that is least burdensome for industry and also for regulators, so we can look at these products and understand, okay, what is being changed, what it remains the same, and what are the processes that are being used at these organizations. And next slide, please. So as many of you may be familiar, we put out a discussion paper, and this was essentially a request for input from the community. The ideas here drew on a number of sources, including the International Medical Device Regulators Forum, work on SAMD, FDA's benefit risk framework, our guidances, including that related to software modifications, and the total product lifecycle approach as envisioned in the PRECERT program. Next slide. I'll mention very quickly what's been touched on previously here today is the idea that oftentimes you do have a human in the loop. So we're looking at a spectrum from left to right of, on the one hand, algorithms that are trained using machine learning, but then locked and updated, at a certain frequency, largely by humans. As we move to the right, in the extreme, a continuously adaptive algorithm that, let's say, every time it comes across new data, it has the potential for changing the way that it functions, and the spectrum in between. And what we said in the white paper is that if you have a good plan in place and good processes in place, if adaptations are pre-specified, then a similar decision-making framework could be applied across the spectrum. Next slide, please. And here is an image from the paper. I'll walk you through it quickly. Again, I think you've seen similar versions of the AI/ML workflow and development cycle. And we'll start here in the shaded box like region at the top that's labeled Good Machine Learning Practices. And this is a development environment where data is used to train a model, which is validated. And then, and this is an iterative process, as we know. If you follow those two arrows toward the bottom of the slide, the white and green together, that's a model being deployed into the market, so to speak, where new data is inputted into the model, and the output on the right is monitored, and that should inform the development of the model and the algorithm itself. And I'm going to place on top of this system the total product lifecycle approach that we propose. And if you could advance the next slide, please, to oversee these technologies. And I'll walk through this pretty quickly, but if you follow the numbers, the numbered octagons in blue from 1 to 4, where 1 is looking for that culture of quality and organizational excellence, these good machine learning practices for these types of products, as Kevin alluded to, is sort of step 1. Step 2 and 3 is more analogous to a pre-market review, as we have today, especially 2, where the product is submitted with appropriate validation data and intended use, and it's given clearance or approval, or in some fashion marketing authorization. Number 3 here is a review of the plan for the algorithm to learn, and we've called that a review of the SPS and ACP. I'll go into a little more detail today. It's the software pre-specifications and algorithm change protocol. And number 4 is that monitoring of the real-world performance data. Next slide, please. So let's focus for a moment on these good machine learning practices. Next slide. And we've discussed some of them today. They essentially are based on concepts from quality systems, good software development, machine learning, accepted practices in machine learning and artificial intelligence. And there are a number of groups that are working on these in the forms of guidelines, best practices, and standards. Next slide. And I'd like to take a moment, next slide, to focus on this SPS and ACP. Next slide. Where essentially, again, the SPS is proposed to describe the changes, how the software will evolve, and the ACP, if you go on to the next slide, describes the step-by-step how those changes will be made in a way that the product can still be deemed to be safe and effective. And this is in the general categories of data management, retraining, performance evaluation, and then importantly, how will the company be transparent about the changes that have been made? Next slide. I think we're just about at the end of my talk. Just in summary, this is, again, how we see, and you've seen this picture before, we see responsible innovation in AI/ML facilitated by a tailored regulatory framework that allows the evolution of these technologies, both in a clinical setting and in a medical product development setting as well. And I'm going to pause there. Thank you very much.
Thanks a lot, Nassim. It's a very insightful presentation. I'm sure we'll get a lot of questions, I hope, when we read the Q&A. So our next presenter, is Ivan Werthworth from Pfizer. He's the Global Head of Audit and Inspection Quality, and he's going to cover best practices and insight on how to ensure regulatory compliance with validation documentation. Ivan, the floor is yours.
Thank you, Patrick. if we could go ahead and, great. So I'm going to be speaking about GCP compliance of artificial intelligence in clinical trials. If we could go ahead and advance, please, Camilla. So my name is Ivan Walrath, and I head up a function at Pfizer around audit and inspection quality, and as I said, primarily in GCP. If we could go ahead and advance, please. Really, when thinking about artificial intelligence in clinical trial, about what we and preparing for inspection, and understanding the additional risks that need accounted for in establishing compliance. And I really focused on the decision-making aspect of artificial intelligence, which I think is one of the key differentiators there. So I'm going to speak about a little bit about the differences between automations and AI and some of the risks, and then just walk through some thinking about affirming compliance of AI solutions. We go ahead and advance, please. So looking, defending an artificial intelligence system in an inspection, we don't have a whole lot of precedent for it in the GCP environment, at least. And I'd like to start with inspection ready and inspection readiness. And one of the first aspects of that is inspection ready is an adjective. It's not an activity. And it's about having verifiable quality that's based on evidence and documentation of our quality and compliance. And so how do we make that doable and checkable and verifiable in a GCP environment? So we can differentiate the AI from the automation, look at some of the ways of thinking about the risks, and then from that thinking about validating the system. We've heard a number of presentations around this morning, this afternoon, about the system and the validation aspects. And I'm going to spend a little bit more time talking about qualifying the decision making, which is really, I think, the differentiator in this in the AI environment. Can we go ahead and advance, please? Oh, there we go. Okay. So when thinking about automation versus artificial intelligence, so we're thinking about automation or robots or bots mimicking human action or routine work, whether it's retrieving a report, cutting and pasting data, with every activity being explicitly programmed down to the keystroke. It's very process-centric and intended for repetitive tasks. It's very rule-based and very much mimicking a process and the execution of a process or the execution of tasks. We differentiate that from artificial intelligence, where we're simulating intelligence with the machines, reading a report and enabling decision making. We've heard a number of examples today about that. We've heard about training and learning techniques from categorized or labeled data, and having experts or human experts train the machine to either think or infer, again, making decisions like humans do, and then using labeled data to train a machine learning algorithm sufficiently, being able to recognize new data faster, and so and really getting down to the key differentiator being around the decision making. So going to really focus on the decision making. If we could go ahead and advance, please. So thinking about the assessment, what is the level of independence? We've heard an example where we've got human in the loop. How independent is the decision making going to be? What's the content area? Is it related to safety? Is it related to diagnosis? Is it related to the generation of clinical data queries? What is the impact of the errors or mistakes? So if the machine wrongly predicts a query, important? What is the impact of that error? And we think about quality, focusing on errors that matter. What are the errors that matter? What is the impact of those? And what's the expected level of accuracy? I know we heard at least one, or might have been two presentations earlier that did not have a target of 100% accuracy. What is the expected level of accuracy and the needed level of accuracy as part of the risk assessment? And then thinking about the risk in different dimensions or domains. Is there a risk related to the study? Is there a risk related with third parties? So, oftentimes there may be multiple third parties involved in using an artificial intelligence solution. And what does that mean? And what risks does that introduce? Risks related to the execution of a process, as well as the interactions and handoffs. So when we have data coming into the machine, a decision is made, and data is coming out, or a decision is coming out. What about those interaction points and those handoffs as well? And then when we think about decision making, which is traditionally performed by a human with a defined role, with specialized training, with experience, and with a defined level of oversight. These are all things that we think about when qualifying decision makers. I won't spend a lot of time talking about the system validation aspect of it, because I think we've heard a lot about that today. So if we could go ahead and advance. And I want to talk about the qualification of decision making and what some of that documentation might look like. So first, around the risk assessment, the documentation, thinking about inspection and evidence of quality. How are we documenting that risk assessment? What is it? What does it look like? How will we produce that? Is it part of a meeting discussion? Is it part of a playbook? Is it part of a formal procedure? Having that documentation and that reproducibility of that risk assessment. In terms of thinking about it almost in a personification-like lens, we think about the job description of a decision maker. So what is the scope? What are the requirements? What is the documentation of the intent, as well as the expectations around performance? What is the decision making in this in the instance of AI being used? What is the intent? What is the expected performance, the expected level of accuracy, and having that well documented. When it comes to training, if we think about when we're looking at a decision maker, what are the records of training? So we've heard about teach cycles, the curriculum essentially for that training, and the materials. So we've heard about historical data sets, the machine learning process. We've heard about different versions of the models, the statistical models being used that are underpinning the decision-making, the AI. And then the records of those training. Can we document and can we substantiate and reproduce that training, both in terms of the materials that were used as well as the instance of that training, and an evaluation of that training effectiveness. How have we evaluated and tested that the training was effective? We think also about experience. So we think about this the way we might look at a CV. What is the level of consistency of performance to expectation, and that history of performance. And then lastly, the oversight of performance. So what is the documented, the verification that we have of the prediction? Are we using metrics, for example? Do we have documented feedback related to the prediction models that may be feeding back into the teach cycles and those training loops? And do we have a plan? And the plan and evidence of the oversight. So for example, do we have documented review of the performance, where the humans in this case are reviewing the performance, looking at the metrics, looking at all other indicators of performance, and that review being documented. Again, it's then evidence of the oversight. And I put audits there. Certainly an audit is a way of looking at oversight of performance, although audits are, tend to be more retrospective. But in terms of thinking about the qualifying of the decision making, which is really in some ways in addition to the validation of all of the technology and the technical requirements and of the system per se, to ensure that we can qualify and stand behind the decision making. So this is again just a way that I'm thinking about this in terms of upcoming inspections. So thank you, and I can turn it back over to Torsten and Patrick.
Thank you.
Thanks, Ivan.
Thank you, Matthew. Thank you, Ivan, for the two very excellent presentations. And now we come to the Q&A. Given that the time already got way ahead, we wanted to finish originally 25. I will go directly to the general Q&A. Obviously, you can ask questions in general Q&A to the last two presenters as well. So I give the floor to everyone. Raise your hand if you want to ask a question.
And maybe in the meantime, Torsten, I have a question for Matthew. I know, Matthew, the 2019 proposed guidance was the first regulatory document published, and I know you sought feedback from the industry. Could you share some your insight of what feedback you got from the industry about that proposed guidance, Matthew?
Yes. Can you hear me now?
Yep, we can, Matthew.
Perfect. Yeah, thank you for the question. And what we issued last year, just to clarify, it was a discussion paper, and we don't consider it to be guidance yet. But as you mentioned, it was to put out some ideas, to ask questions, and to gather input from the community. We received a large number of comments, on the order of 1,000, which the group here, carefully read, and I can talk about some of the high-level messages. First of all, I think there was a lot of positive response to the idea of a predetermined change control plan, essentially a proactive statement of how algorithms are anticipated to evolve, and we had a lot of interest both within the comments and outside of the comments about utilizing a mechanism like that. We also had a lot of positive feedback about the codification of good machine learning practices, and included in that was multiple requests for FDA to take a role to try to help harmonize these many different efforts among different standards organizations, and to that end, we do actively participate in a number of those standards efforts, and also we participate in some of the collaborative communities. One of them is the Xavier World AI program, where you have stakeholders who are participating in many of these other standards efforts, but explicitly part of the goal is to try to make sure that we don't, in our efforts to standardize things, codify many different approaches. There were some other high points of feedback as well. I think more clarity was requested on, what does transparency mean, and how does a company achieve it with labeling. There was a call for more further development of the framework and delineation of what what we called a focused review would look like, and that was described as if, as Kevin Lyman described, he's looking to evolve his product to a new functionality that was not anticipated initially, how can we expedite the review to focus just on those aspects? So maybe I'll pause there. Those were some of the high-level messages we received. And I should just mention, we're working to respond, through our activities to those suggestions.
Thanks, Matthew.
Janos has raised his hand.
Yeah, hi, can you hear me?
Yes.
Yeah, can you hear me?
Yes, we can.
Yeah, hi. Thanks a lot. Actually, it's an excellent conference, and thanks for the excellent talks. Unfortunately, I was late to come because I was in another conference, so I just caught the last couple of talks. And I wanted to ask 2 questions. First, concerning this, the idea of continuous learning. Are we always referring, when we refer to continuous learning, are we always referring to machine learning algorithms, which after deployed, there will be continuous learning, but not applying their learning on humans in real practice? Or do we see some potential in having, let's say, genuine continuous learning, in which case machine learning algorithms could be indeed continuously learning and applying? And in the second case, what risks do you see, and how do you think it could be protecting such risks, against such risks, if we could ever have such algorithms, in medicine especially? And my second question concerns a bit more liability issues. I'm wondering if you have considered, or what are your ideas about liability, especially in the context where a machine learning algorithm would be working together with a physician and a patient. So in that sense, there will be this triangular relationship. And of course, in this triangular relationship, the physician would be required, or is required under laws of consent, to obtain the informed consent of the patient. And let's assume that the physician has opinion A, the machine comes with opinion B. The physician has to obtain the informed consent of the patient. Do you see any role, to be more concrete, do you see any role of explainability of AI in this context especially? Thanks a lot.
Whom do you asking this question, or is it generally directed to the audience?
Maybe I think it will be more for, I think it was Matthew from the FDA.
Do you like
Sure.
to answer?
Thanks. Yeah, absolutely. So in regard to the first question about continuous learning, and Kevin, feel free to chime in as well. But I was using the term quite broadly to encompass both scenarios which you described. And I think my understanding of the point that Kevin was making is that continuous learning includes incremental learning, and I share the same opinion. Now, I think you asked about how about systems that I would describe as being continuously adaptive, that are adjusting their device functioning algorithm in the field in response to new data. And I would say that broadly, if the proper controls are in place, there is no prohibition against having a device that functions in this manner. But again, we are very interested to hear industry's proposal for providing the right guardrails and controls in a product that would function in that way. In regard to the second question, I don't want to talk too much about liability. It's a complex area, but I can just reiterate a few fundamental principles that I think you can apply to the area, and that is that it's very important that the software is clear in its intended use, and that it is used in a manner in which it was intended. And I think that software that assists a clinician is very different than software that will perform a task independent of a clinician. And I think that has an important impact on liability. But I would just open this up to Kevin. Anything else you want to say about continuous learning?
I'm afraid Kevin has left us.
This is Alan here. I don't think Kevin is with us anymore. So,
okay, I did notice.
Certainly, my impression of both his presentation and Melissa's, they both talked about using the continual learning experiences as a way of improving the testing and the validation of the product, but that they were a gated method for releasing the new training material into each product. So it wasn't a continual learning on a live basis. It's simply a way to improve, which will result in a new release at a certain point in time.
Yeah, I would agree. This is Dimitris. I don't see the point of the continuous learning unless you're applying it, but it's a matter perhaps of controlling the cycles of the learning and applying as opposed to truly live. Maybe that's what you're getting at, Janos. But I would say the scenario of what Prasanna presented earlier that we're doing, it is continuous learning and it's being applied. It's just not on the keystroke. It's in controlled cycles or batches, if you will.
Thank you, Dimitri. Thank you, Alan. I would now allow Christian to ask his question. Christian, please.
Patrick, I guess it's back to you, and Torsten.
No, thank you. Is there any other questions from either a presenter or from anyone in the audience? We got one question, which was how to take education of users into account. Maybe Ivan, do you want to answer that one?
Sorry, I heard it sounded like there were two different people talking, so I couldn't hear.
The question is how to take education of users into account.
Okay, so that would be then the users who are interacting?
Yep. Or validating, I'm guessing. Yep.
I think it's a fundamental aspect of quality and compliance that the users and people who are interacting with the system have the appropriate education, and that needs to be pre-specified. There's an echo, so I'll stop there.
Okay, Torsten and Patrick, I'm being aware of time, so maybe if there's no burning questions left, we want to go to the closing remarks, if that's okay with you.
Absolutely, let's do that.
Absolutely.
Okay, great, and thank you, everybody. I don't know, have you all seen this extremely funny YouTube video on how a virtual meeting would play out if it was physical? So at the beginning of this session, I felt like I was in like a candid camera version of this video. So, but I'm happy to say that the second part of this meeting was running really smoothly, and thanks to everybody to staying online and for some really great presentations. I would like to just make a quick recap of what we've been talking about. So for setting the scene, we heard a lot of purposes for use of artificial intelligence, so in precision medicine, classification of diseases, diagnosis, patient enrollment, data monitoring, medical imaging, pharmacovigilance for auto-classifying adverse events, to increase the usability of real-world data, optimizing trial design, and identifying new molecules of interest. So it's really become apparent that it's more and more something that can be used We saw specific use cases on smart data query use, signal detection in wearable device data, protocol deviation trending, using natural language processing, and also about when randomized clinical trials are not possible, how do we estimate the counterfactual, so the synthetic control arm, use of artificial intelligence in complaint handling, and artificial intelligence for regulatory science as well. So, for instance, tools for diagnostics. Some of the challenges I've noted during the day were things like limitation of current models, necessity of high volume of data for training. So unless you use shallow machine learning and powerful mathematical tools and methods, validation of continuous learning algorithms and how that's integrated into existing data flows. The regulatory framework was mentioned, things like ethics issues, data privacy, false positives and negatives, so sensitivity and specificity, and also replicability and reproducibility is key, as is good practice to ensure that the number of bad decisions are not increasing with the amount of data. So in other words, also garbage in and garbage out, and necessary data quality is necessary but not sufficient on its own. The space where we train the AI is highly critical, and we should be aware of potential bias on estimates depending on the variability of training sets. Training and test data sets should be maintained and traceable. And also, when does it become different from now? So when do we leave the level of comfort? So for instance, when scripts look and feel different and the documentation is more challenging. And we were also hearing about continuous machine learning and some points about when is something abnormal, and then going from that and continuously learning to also now indicate what issues actually, what is the issue, what is wrong, and improve the model accuracy, but also discovering things that are suddenly being approachable due to this. We heard here, last but not least, also in the discussion where Matthew gave a presentation describing the balance that we were talking about early in the day about assurance that the safety and efficacy is maintained versus the burden of reducing the burden. And we heard the workflow FDA has, and that was an inspiration, I would say. And also, Ivan was talking about an encouragement to overcome the fear of using and defending and inspecting artificial intelligence just because, we don't have necessarily all the experience we think we should have. And differentiating artificial intelligence from automation, and also remembering to the whole way assess the risk, validate the systems, and qualify the decision making. So those were some of the points I was taking home, at least. And I think today has really been an exercise in building trust, and it's really important to continue collaboration on all levels, so among regulators, researchers, industry, and patients, and all hopefully for the mutual benefit of a smoother transition for new technology, and specifically where data integrity and patient safety is not negatively impacted. And ultimately maybe leading to higher acceptability when the AI is implemented in clinical trials, and really depending on the definition, it's already here. And we need to have artificial intelligence implemented in a way that ensures that the clinical trial is a trial where the efficacy and or safety of the medicine is tested, and not a trial where we're simultaneously testing an AI algorithm. So we need to be able to trust the data from the systems, and therefore it's essential that system functionality is adequately validated and tested and documented. And in that context, transparency and verifiability is also key. I think today was an important first step on the way, and I think some of the things we're going to discuss going forward. Also some of the gray zones. We heard Matthew talk about medical devices, and we are here as GCP inspectors, so we would be more looking at the GCP framework, but there's no doubt that there's a huge overlap here when it comes to artificial intelligence, how to adhere to ethics requirements and to the GDPR as well. And I think we would, at least in the Danish Medicines Agency, be keen to see some pilots, and I'm sure that the other agencies will as well. So I can only say that it's a good idea to approach regulators, maybe also for scientific advice. And I hope you'll come with feedback also and input to future meetings that we hope will be possible to have in 2021. And I think that the first point is obviously to make sure technology is working from the beginning of the meeting. And before I give the word to Jane, I would just like to finally thank all of you, and especially the speakers and the moderators. Thank you very much for driving this discussion, and to the program committee who's been meeting a lot of times to have this organized. So people from FDA, the GCP Inspectors Working Group, and the ESOP Group, the SCDM and eClinical Forum and UCROF representative. And last but not least, a great thanks to EMA for hosting this, and for Camelia for a huge effort in making this run. And I know there's been a lot of fire distinguishing going on behind the scenes today, and I'm sure that everybody are really, really tired, and thank you for solving the problems that we had initially. So, Jane, I would hand it over to you for the final remarks.
So from the EMA's perspective, yes, I can offer a sincere apology to the speakers and to participants really for the technical issues. I can say a lot of work went into the preparation of this meeting, but this is for us to debrief afterwards. The speakers were valiant in their resilience, and I would applaud that. So thank you for your patience with that. From the AI perspective, it's clear that this features in the strategic goals of the agency and the medicine network. It's a sincere wish that from the EMA that innovation is translated into new medicines for patients in a way also that that protects patients, and the data that comes are robust and reliable. So how do we make this happen? I think some of the key issues really are to continue this dialogue and to consider, well, do we need guidance, for example? But I think it's a very dynamic area. Any guidance that has developed has to be responsive and agile, and really build on real experiences that can be developed. It's very much not just a GCP issue, but cuts across many areas. And reaching out today to all of the collaborators was very important. We have industry associations and in small and large industries, CROs. We have regulators from other agencies, the Commission, the NCAs and the inspectors, but also we have patients and academics and healthcare professionals. So I think the collaboration is very important as we go forward. I think, yes, the capacity and the capability in the network is important, and that's also for us to reflect on. But I think really one of the most important things to build experience in this area for everybody is for concrete cases to come forward for dialogue with regulators in the context of scientific advice and in the context of the Innovation Task Force, so that we can build on that. Very much a big thank you to everybody in terms of the organisation of the meeting. A lot of work has gone into this, and I look forward to further discussions. Thank you very much. I think we can now close the meeting. And a good evening.
Good night, and thank you, everyone.